You're listening to the Common Descent Podcast.
Hello, Will.
Hello, David.
Hello, everybody out there, and welcome to Common Descent, a podcast about paleontology, evolution, life history, and so on. This is episode 195. Longtime listeners of the podcast will know that 5 at the end means it's a plants episode.
Woo!
This episode, our topic is leaves. Very important piece of plant functions.
Plant tech.
Plant tech. This is a very key part of plants. Now, it is the middle, we're in croc and snake month territory, and when we looked at the schedule, usually, the last couple of years we've done croc and snake month, we did a croc-themed episode for croc month and then a snake-themed episode for snake month, and they were back-to-back. This year, the 5 episode falls right in between them.
Yep.
So there will be a snake-themed episode. Stay tuned next episode. We'll talk more about snakes, but we got to put a plants episode right there in the middle, and because we will be talking about plants, and because we will, of course, as always, be joined by our favorite paleobotanist, Dr. Ali Baumgartner, our plant person, to talk with us about plants after we get through the news and such to the main discussion. We thought that since you're talking about crocs last episode, and I'm talking about snakes next episode, we might as well give Ali the chance to talk about her favorite thing, and that's leaves.
Yes, it is.
This is Allie's whole deal. Allie loves leaves. Allie's a leaf person. We will talk about what leaves are, how they function, why they're important, what they mean for us in the fossil record, both in terms of what they tell us about the evolutionary history of plants, but also how leaves themselves are important for understanding the fossil record. We'll go into all those different aspects of leaf anatomy and evolution and history, because it's super cool.
Oh, yeah.
Very exciting topic. Allie, very excited to talk about this. Also, requested.
Yay.
This particular topic was requested by Jackie, James, Taterboy, and Dr. Jazz. Thank you, everybody, for those requests. Hey, some announcements before we get into the rest of the stuff. Here at Common Descent, we are supported wholly all the way by our supporters on Patreon. If you hop down to the episode description of this episode, you will find a link to our Patreon, or you can go to our website and there'll be a link to the Patreon. From there, there are several tiers at which you can support us financially here on the podcast. You get all sorts of goodies for supporting us, including at certain levels, shouting out your name here on the podcast. We got a bunch of new patrons to shout out this time. Welcome to our newest patrons, Jarrett, Michael, Elise, Full Sail, and Carboniferous.
Welcome.
I didn't think about this when I put that on there, but Carboniferous is an excellent name to shout out on a plants episode.
Yes, it is.
That works out pretty well. Hey, if you would like to be a patron and support us, you get access to bonus content. We do bonus news. We do live streams with our patrons at the higher levels. You get stuff like t-shirts, and you can hang out with us. You can get a hangout, personal one-on-one with us at the high levels of Patreon subscription. And not only that, but now in the summer is maybe the best time to join us on Patreon, because it is, as of the release of this episode, Snake Month.
Woo!
This is the first episode of Snake Month. Every summer in June and July, we celebrate Croc Month in June and Snake Month in July. Croc Month is over. Thank goodness we got that out of the way. And it is time to celebrate snakes.
Rude.
We have all sorts of snake. The next episode is going to be snake stuff. We've got the Snake Stuff channel open on our Discord only for the month of July for all of your snake conversation needs. On the 16th of July, we will be having a Snake Month live stream open to anybody who wants to join on our YouTube, just like we did for Crocs last month. Get on down to the episode description, go to our website, and you will see information for those things. But also on top of all that, during June and July, there is a special tier on Patreon, the Snakes and Crocs tier. If you join at those tiers, you get a bunch of goodies. You get special Croc and Snake Month stickers, and subscriptions that we receive at the Snakes and Crocs tier during June and July will contribute to charitable donations we will be making towards reptile conservation at the end of the summer. Your subscription to us at those tiers this year not only supports us in our science education efforts, but we will be taking a bunch of that money and donating it to the Crocodile Research Coalition, Save the Snakes, and this year, Turtle Survival Alliance, Because we're looping in some other reptiles this time. Because other reptiles are also cool. We don't do a turtle month here on the podcast, but turtles are pretty cool too, I guess. And not only is the summer a great time to join us on Patreon, but the summer of 2024 is an even better time than usual to join us because a generous fan of the podcast has agreed to match donations for this year's Croc and Snake Month up to $1,000. Amazing. Fantastic. That fan is making those donations on behalf of Mayflower Enterprises. This is the part where we talk them up because they're doing such a cool, nice thing.
Yeah.
Mayflower Enterprises is a small engineering firm in northern Indiana, here to help you discover your potential, capital D-Y-N-P.
Mm-hmm.
Whether you're a newly founded startup, been in business for 20 years, or just have an idea on a bar napkin, Mayflower stands ready to bring their Fortune 100 experience from automotive, aerospace, manufacturing, and construction to help your organization discover its potential through process development, personnel training, future-proofing, and more. They have a passion for the environment, scientific discovery, and deeply nerdy hobbies that include listening to the incredible team at the Common Descent podcast. They wrote that.
Yes.
They wrote that.
Their words, not ours.
Now it's our words. We are the ones saying them. We are so thrilled they continue to partner with them this year to help support Croc and Snake Month by matching funds for the Crocs and Snakes tiers up to the first $1,000. Amazing.
It's really cool.
Thank you so much to our generous donors. Thank you to all of our patrons who support us. Thank you to all of our fans who support us. We hope you'll get involved and engaged for Croc and Snake Month this year, especially now that we're in the second half, which is the best part. We're in my month now. You gotta put. I let you get away with all sorts of.
I'm not disagreeing, but I won't agree
with
either.
all sorts of croc talk. Hey, also speaking of special cool stuff that we do, when this episode comes out, we will have very recently released the next episode of our Spotlight series.
Yeah,
Spotlight 2024 coming out all throughout the year of 2024. New episode beginning the first Wednesday of every month, where we sit down with other podcasters in paleontology and related stuff and talk about podcasting and science communication. Here at the beginning of July, we just released an episode with Travis Holland, who runs the podcast Fossils and Fiction.
That was a really interesting talk.
Yeah.
I learned a lot about what Travis does and the kind of research they do.
It was very cool. Also, our second chat with an Australian for our Spotlight series, which is more than I expected to have. That's great. This is fantastic. We're shining a light all the way on the other side of the world. Those are some announcements from the world of Common Descent, what's going on with us. And now let us move on to talking about what's going on in the world of broader sciences. We've got some news. Every episode we talk about what's new, new research, new publications and reports from the science of paleontology and related stuff. Will, now it's your turn to talk a bunch.
Woo! My first news is about a new species found from Brazil of pseudosuchian.
That's very croc-like, and
I can't help when this news came out.
It's a little bit. It's a little.
I didn't write
inconsiderate.
I didn't write it.
That it's Snake Month.
I'm simply reporting it. I'm simply speaking about this, and this isn't the snake-themed episode yet.
Next episode, just you wait. There's going to be so many snakes.
Not even allowed to mention things with legs.
Can't even mention nothing with legs. No talk of anything with legs. All right, tell us about your almost croc thing.
This is research by Rodrigo Mueller in Scientific Reports, and the article is by Paul Smaglik in Discover Magazine.
Go to the blog post for this episode to find the links to these articles.
So, back in the Triassic, before dinosaurs and pterosaurs had taken over and were the dominant groups, the main land reptiles were pseudosuchians. These were your big predators that were roaming the land. These are croc-like animals. They're on the same side of archosaurs as the crocodilians, but they are before most of the groups that we start calling crocish.
Yeah, this is the extended family of crocodilians.
And they predate a lot of the lineages that you think of as ancient crocs. These are typically terrestrial, standing mostly upright with like longer legs. So they look like a croc if you stretched it up. If you took the dimensions and stretched it up. They've got taller faces made for moving around on land. They include some of the largest predators of that time. But not all were large predators. There were smaller members of this group. The Gracillus suchidae were members of this group that were small, gracile, as the name suggests.
Sure, slender.
Slender, good for moving quickly, probably. Often under a meter in length. So actually quite small. They've been known from China and Argentina mostly, with some maybe evidence from Brazil. This new fossil is the first unambiguous member from Brazil.
All right.
This new species is named Parvosuchus aurelioi. It is from the Dinodontosaurus assemblage zone in Brazil and is about 237 million years old. It is composed of a complete cranium, so we got the whole skull basically, vertebrae, pelvic girdle, so we got the hips and everything, and hind limbs.
Oh, nice. So that's most of the animal.
This is a decently preserved individual. Phylogenetic analysis places it between two species. One is Gracillosuchus, so it is squarely within this group. It evidently helps fill up the taxonomic bracket of Brazilian pseudosuchians in general, so it is helping fill out this overall group of pseudosuchians in this area. It's not the only pseudosuchian known from this site, but it is the smallest by far. Its skull measures just under 6 inches long.
A little guy.
This is very small. Estimated that the entire skeleton would have been about 3 feet long. So it's right within that meter length that we were talking about.
Yeah. And I bet a bunch of that's tail.
Oh, yeah, absolutely. Like, today's crocs, about half the length is tail.
Yep.
So probably a pretty small little body with just a little half-foot face. This indicates that it was almost certainly not the apex predator in its environment.
Probably not.
Its skull...
Bugs were terrible.
Right, yes. For everything at grass level, which wasn't there yet, so...
Right, yeah, exactly. Nowhere to hide.
The skull is slender and has pointed teeth that are curved back. So, think of kind of like a monitor lizard is what I was probably a decent picture. This is exciting just because it is first member from this group from this area that is undeniably in the Gracillosuchidae. It also is demonstrating the diversity of pseudosuchians in this site. There are much larger members found alongside this species. Prestosuchus is one that's known from this site and is a much larger, much closer to being an apex predator. So it's showing the diversity of niches that pseudosuchians would have been occupying within the same habitat, which gives a better look at what the environment and ecosystem, the food chain would have been like during the Triassic, and is just helping us to understand the full diversity of pseudosuchians at this time.
Yeah, it's nice to be reminded of the diversity of these ancient groups. We so often think about dinosaurs and pterosaurs, and before them pseudosuchians, and modern mammals, the groups of animals that become widespread and the most important things across their ecosystems. We think of large size as one of the signs. This is a group that has made it.
Yes.
And now you are huge and you're unmissable in your environment. And that's true. All those groups do reach large sizes. But it's nice to be reminded that one of the signs of that success is diversity of forms.
Mm-hmm.
Mammals today, even, big predators like cats or canids, they range in size from, bobcat-sized all the way up to tiger-sized. It's nice to be reminded that, the pseudosuchians were successful not just because they were big apex predators, but because they occupied a bunch of different niches in their habitats.
And it's also, I think, good to be reminded of these smaller members because, as is often the case with even monad groups, but especially fossil groups, the big members are the ones that become famous.
Yes.
Like Postosuchus, which is like the famous one from Walking with Dinosaurs, is one of the biggest members of this group and was one of the largest pseudosuchians that ever lived. But they weren't all that big. But when you only hear about the big ones, it's very easy to forget that there were ones that were the size of your cat, that were dog-sized and would have been filling that fox and small predator role in the ecosystem.
For sure. Very cool find. Well, now that we've finished with your blasphemy...
It's just, the slight overflow.
Bringing archosaurs into my snake month. My first bit of news is also about archosaurs. I've got, this one's a dinosaur. It is not at all like a snake. This is research about another new species of archosaur. This one is a horned dinosaur named Lokiceratops.
I like it.
This is a super cool find.
I like the name.
This is research published by Mark Lowen et al. In the journal PeerJ, and in the blog post we will link to an article in Science Alert by Michelle Starr. Lokiceratops rangiformis is the new genus and species of horned dinosaur. This is your Triceratops-esque group of dinosaurs. Episode 87. Lokiceratops comes from the famous Judith River Formation in Montana, which is way up north in Montana. Dates to the late Cretaceous around 78 million years ago. Lokiceratops is known from some very nice remains, including most of the cranium, not unlike your discovery. It looks like the mandible, the lower jaw, is all or mostly not preserved.
Okay.
But the rest of the skull, the frill, the horns, the face, is largely preserved, which is not unusual for ceratopsians.
They have a very robust face.
That's the part you're going to get if you're going to find them. And it also looks like there's some bits of shoulder and pelvis and a few vertebrae, so there's a smattering of the body as well. This provides a very good look at the head, which is handy because that's how also we identify a lot of ceratopsians.
Yep.
Now, if you remember episode 87, which is the episode where one of us sounds like we're underwater, the late Cretaceous horned dinosaurs come in two main varieties. Centrosaurines, which includes Centrosaurus and a bunch of other, Pachyrhinosaurus is in that group, and the Chasmosaurines, which includes Chasmosaurs and Triceratops and a bunch of others like those. The skull features of Lokiceratops place it firmly within the Centrosaurs, so not the Triceratops side of the tree, the other side of the tree. The skull's full length is likely over 2 meters long.
Ridiculous.
Huge head.
They just have such big heads.
And its total body length is estimated to have probably been about 6.5 meters or more, 20 feet or more, which makes it not quite as big as Triceratops, but among the biggest of the Centrosaurine horned dinosaurs.
Yeah.
Two things that really stand out about Lokiceratops in this discussion. First is the head. Every time there's a new species of ceratopsian, we get a new arrangement of horns and frills.
Yep.
This is the thing that they do. Every species has a different set of horns and frills. Lokiceratops has two big brow horns, so the horns sticking out over the eyes, like Triceratops does. The frill has little spiky bumps around the edges of it. At the top of the frill, right up in the middle, are two big spikes that stick upwards, and then two sort of horn-like projections that kind of curve off to the side a little bit. They look almost like ram's horns.
Yeah.
A little bit curving off to the side. Now, in the paper, they simply say that it's named Lokiceratops after the Norse god from mythology, but presumably they are naming it Lokiceratops because its horns look like Loki's helmet from the Marvel movies.
Which, he has a horn helmet in the old myths as well.
Right, so it might be, charitably, we might say that it's a reference to that. The species name Rangiformis is Formis, the form of Rangifer, which is the genus of caribou.
Okay, nice.
And they name it that not only because, the frill and the horns are probably being used similarly to antlers for showing off and for perhaps combative displays, episode 140, but also the frill, horns, and spikes up on the top are asymmetrical.
Oh.
Of those two big spikes, one of them seems to be noticeably larger than the other one, and it looks like the curved horn parts are also somewhat asymmetrical. This is seen in several species of ceratopsians. This apparently is not something new. And you also see it in deer and animals with antlers, where their display structure is different on the left and the right. Apparently, according to the paper, Lokiceratops's frill stuff are noticeably asymmetrical, like a little bit more asymmetrical than we typically see, which apparently is also true of caribou.
Okay, yeah.
That's a thing in caribou. They are a little extra asymmetrical. So the name Rangiformis is in reference to the fact that it's got a weird horn situation, kind of like caribou do.
Cool. Oh, I like that a lot.
The other thing that's interesting about Lokiceratops is where it comes from. It comes from the Judith River Formation in Montana, which is the same age and very close in geography to the Oldman Formation in Canada. Between those two formations, this now makes 5 different species of horned dinosaurs known from this one small region all at the same time.
Yeah.
Which is a lot of species of this one group of dinosaurs that are presumably living very similar lifestyles in the same place. The phylogeny identifies Lokiceratops as closely related to Albertoceratops and Medusaceratops, which are also known from this general region at a similar time, which suggests that this was a radiation of closely related species that stuck around in one geographic area. They radiated, but didn't spread out very far. They stayed mostly in one place.
Yeah.
Which the authors list at least 3 other examples of that in ceratopsians in Western North America in the late Cretaceous. That this seems to be a thing that happened repeatedly, that you got these little regionally limited radiations of new species, thus resulting in high regional endemism.
Yes.
That is, you have a handful of species of horned dinosaurs that are found only in one particular part of the continent. They suggest that this could mean that they were speciating rapidly, that we were just getting lots of new species and there wasn't as much availability for them to spread out. If they were exhibiting high speciation rates, that might be related to environmental conditions. It could be a side effect of sexual selection, which we might expect to see quite a bit in animals with such wild display features. But they also point out that this could mean dinosaurs were more diverse than we tend to think about them.
Yeah.
If you had a bunch of species that were relatively limited in their geographic range, this is different from what we see with large mammals today. A lot of large mammals today have very wide ranges across whatever continent they live on. It could be that dinosaurs were exhibiting ranges differently than large animals that we see today.
Yeah.
You might have had regional pockets with several species of large dinosaur that were only found in one particular relatively small region.
Yes. Very interesting.
So there's a lot going on with this discovery of, it's not just the cool horns.
Yes.
There's a bunch of cool implications here.
And questions that have been presented. And ceratopsians, there's always a bunch of interesting questions because their frills and their horns are so ornate and bizarre and differentiated that, we talked about that in the episode a whole bunch of, is this for combat? Is this for species recognition? Is this for display? Is it for all 3? Something important is happening with these because they're so selected for, obviously. But exactly what isn't for sure clear. And then when you get something where it's like, also, some of them, like this one, have notably asymmetrical horns and frills going on. All right, so what's going on there? Is that how you interlock when you fight each other? That right. You're all asymmetrical roughly the same way, so when you interlock, it's like a handshake. Yep.
Is it just that this one individual was just a little extra asymmetrical for some reason?
Were you using... Because I know with like caribou, some of the asymmetric is that they have a part that comes forward for sweeping snow out of the way. And it's usually only on one antler, to my knowledge. This snow shovel thing. Were you using them in a way? Are they for like scraping against trees? Do you display by scraping against trees?
Are you displaying like cuttlefish where you're displaying to one someone on your left side and someone else on the right side?
Yeah, you line up between rival and female and you're displaying differently.
And then there's the added layer of the relationships of this dinosaur make it really interesting, and where it's found makes it really interesting. It's cool to find a single fossil that has the potential to teach us about so many different facets of these dinosaurs.
Yes.
This is one skeleton that is burdened with glorious purpose. I did it. That's a thing that famously Loki said in Norse mythology.
Yes. In modern Norse mythology. Yes. Very cool. Well, my next news is kind of not about a ceratopsian. This is a research that looked into whether or not it is accurate to say that Protoceratops inspired the myth of the griffin.
Oh, so
And-
the tie-in is mythology.
Yeah, and they found that probably not. So
Okay.
it's not really about Protoceratops.
Right.
But we have to talk about Protoceratops. This is research by Mark Witten and Richard Hing in Science Reviews, and the article is by Evrim Yazgan in Cosmos magazine. So there is a fairly well-known idea that the griffin from Greek and medieval mythology and others as well, goes all the way back to Egyptian and Middle Eastern art, the creature that is the front portion of an eagle and the back portion of a lion with two large wings, was inspired by remains of Protoceratops, which is a hornless ceratopsian. So it has a frill, no horns, but big beak and robust little body. One of my favorite dinosaurs. I love
Very
Protoceratops.
In my Magic School Bus computer game, there was a mini game where you had to be a little Protoceratops and fight off the oviraptors who were coming to steal your eggs.
Yeah!
It was pretty fun. That's terrible for the oviraptors.
Yes.
Who were not stealing eggs.
Yes, that is
as it turned out.
Protoceratops propaganda. Protoceratops is known from Mongolia.
Protoceratopsaganda. We did it. We got there. It's going to be a long episode.
Yep. They are known from Mongolia and China, and this link between them has been around for a little while, and is one of the more well-known of these. There are others of dragons and being inspired by dinosaur bones.
Sure, the Cyclops.
And the elephant.
Bit by like mammoth or elephant skulls.
Absolutely. There's a bunch of these kinds of stories, and we've talked about them before in other sections of the podcast, that while some of them could absolutely be possible, there's not actually direct proof for a lot of them being the direct inspiration for these mythical creatures.
Right, and it's a very hard thing to draw a for sure line between.
Yes. So they wanted to look in, does this hold any water? Is it reasonable? Is really what they're trying to determine. This idea first came around in 1989 when folklorist Adrian Mayer wrote a paper suggesting the link, and then followed up in a book, The First Fossil Hunters, in 2000.
Mm-hmm. Very popular book, and I think that seems like around the time that this became much more of a widespread idea.
Yep. The idea is that the remains of Protoceratops found in Mongolia and China were found by people going and looking for gold deposits in those areas, and then finding fossils or being shown fossils, and then bringing those stories back with them southwestward along trade routes, and eventually ending up in ancient Greece, where a lot of the myths first gained wide notoriety. According to this paper, they find this uncompelling for a few reasons. One, it does ignore established histories of griffin art and myths. Like I said, they go back to ancient Egypt.
Right. Places without Protoceratops.
Without Protoceratops, and that don't follow this link made between China and Greece. So there are other evidences for the griffin that don't seem to line up with this explanation. There often has been made the link between them looking for gold deposits and finding Protoceratops, and then the gold hoarding and protecting behaviors in some of the stories of griffins.
Mm-hmm.
But the nearest deposits that have Protoceratops are hundreds of kilometers away from the nearest gold deposits.
Right.
So
we were in the wrong place.
So it doesn't seem like if they indeed were finding gold, they shouldn't have been able to find Protoceratops. So that doesn't seem to actually be a close association between the two. So it doesn't make sense that they would have stumbled upon both of them. Along the note of them noticing fossils, they make the point that dinosaur fossils are not an easy thing to notice. So just right on the face of it, that you're usually only going to see a portion of the bone sticking out of the rock, and unless you pursue it, you're not likely to just see the Protoceratops skull laying on the surface.
Right, and even then, they're usually not whole skeletons.
Yes.
They're usually all jumbled up and hard to recognize right away.
So it's not that ancient people couldn't be finding fossils, but it still was probably a very rare thing to happen if it was still in the ground, just because dinosaur fossils are hard to find and recognize unless you are looking for dinosaur fossils. And just the fact that the anatomical similarities are pretty sparse.
Mm-hmm.
4 legs and a beak is really all there is. Maybe the frill could be argued that it has an association with the wings, that there's a large flat area. but that there's not actually a ton of similarity between the two, other than that they're quadrupedal animals with a beak. They suggest a much simpler explanation is more likely. They say griffin as a creature in its origins and mythology is consistent with other imaginary beasts that are just chimeras of known animals.
Inspired by animals that you've seen.
Exactly.
And are putting pieces together.
That you looked at an eagle and a lion and you went, boy, those are impressive animals. If together they would be all the more impressive. Minotaurs, chimeras, literally the chimera.
Yes.
We go over this over and over again in the dragons, spooky episodes.
I was going to say, if you hadn't brought up spooky, I was going to mention spooky. This is a recurring theme.
Chimerism is a very common tool in making creatures for stories in ancient and even modern times. Mm-hmm. And that invoking the role of dinosaurs in this lore seems to unnecessarily complicate the origins of the griffin and what is a far more Occam's razor, more likely scenario of it was created through the typical mixing of animals. They make a point to say both that not all of these myths, which they call geomyths, where a mythical creature is connected to a fossil organism, not all mythical creatures need an explanation like that. So those ones we list off before, there's no actual direct evidence for a lot of those. It just makes sense when it's presented to a degree. So it has gained traction, but there's not actually documentation of someone saying they found a skull, and then when we see the skull, we go, that looks like an elephant skull.
Right.
They also make sure to say that there are situations where that seems to have happened. The dragon's tongues being shark's teeth and stuff like that. And there are situations where it seems that they have been inspiration, but that we need to be more careful and make sure that when we attribute it, there's actually some more solid basis to attribute these things to one another. Otherwise, it's just speculation.
Right. Yeah, this is we talked a bunch about this in the Spooky Dragons episodes this past year in Spooky. This is an idea that's been around for a while. The idea of there were people found fossils and then invented mythical creatures to sort of fit the fossils, and it's always been a bit unconvincing because it's very satisfying narratively, but a very difficult thing to actually find support for. The idea of, like we said, elephants' skulls being the inspiration for cyclopses or dinosaurs being the inspiration for dragons. We absolutely have cases where people find fossils and then link it to existing mythological creatures.
Exactly.
Like the dragon tongues, where it's like, oh yes, we already have an idea of dragons. Here are some bones in the ground. These must be linked to those dragons, or dinosaur bones being linked to giant humans. So we have this idea of giants. Here is a big bone that we link to giants. But the idea that people saw fossils and then used them to invent a mythological creature is not only a little bit... It's the kind of stories that's a little too convenient.
Yes.
It's a little just so...
A little too narrative.
It's a little hard to support. And it also, in a real way, at its worst, can diminish the real cultural histories of those legends. It simplifies them and narrows the focus and narrows the significance of it in a way that is very satisfying for us to sort of hear about and speculate about. But it also sort of takes away a lot of the reality of those situations, is that these were coming out of people's imaginations and stories and culturally shared stories for however long.
Well, and they made the point that it also is conflating these ancient peoples with ourselves and assuming they had the same fascination with ancient creatures and their remains as we have.
And the same ability to recognize what they were, which we know they didn't in many cases.
So that it very likely is stemming from a misunderstanding or misrepresentation of what those people were doing and how they were viewing the world.
Yeah. Well, one more bit of news. This one is about animals without legs. Finally, they're not snakes. This is about fish, which isn't closer. But yeah, the idea, I guess, is there. This is about the evolution of fish brains, as informed by some fossil fish brains.
Cool.
This is research by Rodrigo Figueroa et al. In Current Biology, and we will be linking to a press release in Futurity from the University of Michigan. The evolution of brains is very significant and interesting and important for us to understand. Episode 121, we talked all about brains, but mostly we know about brain evolution from modern animals because brains tend not to fossilize.
They are particularly squishy.
There are, however, exceptions. This study examines preserved brain tissue in Paleozoic fish from Brazil. So we're back to Brazil.
Hey!
These fish fossils date to around the Carboniferous to Permian boundary, around 299 million years ago, way back in the late Paleozoic. The researchers CT scanned 8 fish specimens and found a whole bunch of soft tissues. They got brain tissues as well as partial remains of eyes, hearts, and gill filaments. So a bunch of really nice soft tissue organ remains. These brain tissues, which are the focus here of the study, are among the oldest known preserved brains in the fossil record. They are not the oldest. A study last year in 2023, which was published by at least some of the same authors, identified the oldest fossil brains. Another fish called Coxocephalus, which is from England and is about 318 million years old. We talked about it in bonus news, which I went and looked up because while I was reading about this, I was like, so much of this is familiar. Did we do this news already?
Yep.
And I had to go find it. Coxocephalus, the one from the UK, plus these newly scanned fish are all ray-finned fish. That's the fish that includes most of our modern fish, the fish that have rays that support their fins.
Yeah, the see-through fins that you typically think of.
Yes. This study provides some new fish to compare with that other fish and modern fish, but also with this now growing variety of late Paleozoic fish brains, we have the potential to get a better sense of the early condition of what these brains look like. Among the newly scanned fish, they identify 2 distinct types of fish, 2 species effectively, that they're looking at among these fish. The 2 new species and Coxocephalus, the oldest one, have a lot in common. A bunch of features that are the familiar features we see in modern ray-finned fish, as well as some ancestral features that have since been lost. They all share things like, for example, the forebrain is relatively small. They also mention that in all of them, the brain takes up a relatively small portion of the actual cavity in the skull.
Yeah.
It's not taking up the whole space, like in mammals especially do that.
Yep.
They noted that of the 2 new types of fish they found, one of them is identified as closer to modern ray-finned fish.
Okay.
So it's a little bit closer. The other one is a little more of a distant cousin, as is Coxocephalus, a little more distantly related. This is notable because that slightly closer one has an unusual feature of the brain that is shared with modern ray-finned fish.
Ooh.
This is specifically the telencephalon, which is, I think, the front part of the forebrain. Mm-hmm. So the frontest part of the brain. In Coxocephalus and the other new fish, the 2 sort of more distantly related ones, the telencephalon is described as evaginated, which means that the tissues, the brain tissue of the telencephalon, surrounds a cavity on the inside. Whereas modern ray-finned fish have an everted telencephalon, which is the opposite. It's inside out, basically.
Yeah.
The cavity partially surrounds the brain tissue.
Okay.
So the structure has kind of flipped. Instead of having a cavity within all the brain tissue, it is surrounding the brain tissue.
Huh.
This is also the shape that is seen in that other of the new fish species scanned here. So we have a sense of where along the phylogenetic tree that feature of the brain that now we see in all the ray-finned fish first showed up.
Awesome.
So, all together, these ancient fish brains hint at what sort of features were probably present in the very earliest ray-finned fish, and then what features showed up later, and where and when they might have shown up. The authors point out that there's a bunch of other major changes that happened from ancestral ray fin fish to modern ones that we don't have a clear image yet of when exactly all those changes happened. They also pointed out that all those other tissues that are preserved, the eye tissues and hearts and stuff, they said here those are not preserved well enough in those fish for really good insights into their evolution, at least in these authors' interpretation of them.
Mm-hmm.
But they point out that we seem to be a little bit on a roll finding late Paleozoic fish with well-preserved soft tissues. So if we can find more fossils like this, we might get more insights into the evolution, not only of more of how fish brains came to be, and then from there, all the fish that we have today, but also all those other cool tissues in fish that we typically have to rely on modern studies to understand.
Yeah, very cool. It is super neat that they were able to, that there was a feature that with these specimens showed a flip over to the more ray fin fish layout.
Yeah, the format of the brain changed.
Yes.
Which is pretty cool.
And finding stuff like that is cool because it's like, all right, now we have at least by blank time that had happened. But it also can show us stuff of like, all right, that feature showed up before these other features.
Yes.
Of the ray fin fish brain. And sometimes that can be very significant to understanding like, cool, that feature does these things in the brain. So that was more important or preceded the other changes that were to follow.
Yeah. I didn't see any discussion here about what that change in the brain shape would mean for the fish. Or if there's anything known that is like specifically that means this. But it would be interesting as we get more information about their evolution. Do we see other changes around that same time? Like, does their ecology change or like their reproduction change to give us a sense of, did that change in the brain structure allow them to do something new? Was that change in the brain structure a side effect of some other thing that happened? What did that mean for the fish?
Absolutely. Very cool.
Pretty cool stuff. Well, not only did we go through the entire news in this episode without talking about snakes, which is a real shame, but don't worry, there'll be more. We also went through the entire news so far without talking about plants.
Yep.
And that's okay, because the entire rest of the episode is going to be all about plants. After a short musical interlude, we will be joined by our friendly neighborhood paleobotanist, Dr. Allie Baumgartner, who is here to tell us as much as we will listen to about her favorite part of plants, leaves. Don't go away. Hello, Allie.
Hello, David.
Hello, Allie.
Hello, Will.
We're all saying hello. We're all saying it's making sure it happens.
We're all friends here.
Allie, welcome back to the podcast. We are excited to have you join us for this episode, though I hazard to guess we are not nearly as excited as you are.
I'm coming apart at the seams with excitement. I'm just vibrating apart. I've been looking forward to this for a while.
So I guess before we do the topic, the question that I'll ask you to explain to our listeners is, why is our friendly neighborhood paleobotanist, Dr. Allie Baumgartner, so in particularly excited to do an episode about leaves?
Oh my goodness. If you go to my social media, I describe myself as a nerd about leaf shape. That is specifically what I am. Leaves are my favorite part of plants, bar none. There's like, they are what I study. Oh my goodness. I am too excited to even explain to you why I'm so excited about leaves. I love them.
This is an interesting aspect where we have set you, Allie, as our plant expert for the podcast, which means that you come on whenever we talk episode topics about plants, which has put you in the position of representing an entire phylum of life and no, an entire kingdom of life, I'm sorry. And an entire field of science.
Correct.
But of course, within that field of science, most scientists have specialties. And up until now, we have not actually done an episode on your specialty. This episode 195 happens to fall, because of the way the schedule works out, in between when Will and I are doing our themed Croc Month and Snake Month episodes.
Yes.
So an episode in between that, because it's an episode that ends in 5, it seemed very fitting that we let you talk about your favorite and specialty thing.
Oh my goodness. I can't even tell you. We have talked about this for a while, and every time it's like, okay, but Allie, you got to, we can't let you go all out on this because y'all, I could talk about this for days.
There's a whole side. This
Yes.
episode will spin off a side podcast that's
Yes.
just Allie's favorite leaves.
Leave it to Allie.
Oh,
Leave
my,
it to Allie.
Oh, that's so good. All right, I'll
There you
put a pin
go.
in that. That's very good.
Well, let's begin, as always, at the beginning with my favorite category of very simple question. Allie, what's a leaf?
Because I'm lost.
Honestly, this might be the easiest of the simple questions to actually answer, which is delightful because most people do honestly have a pretty good handle on what a leaf is. So to get very pedantic about it, they are the primary photosynthetic organ of vascular plants. Only vascular plants have true leaves.
Cool.
They are typically green and they are typically flattened, blade-like, but broadly speaking, they are one of the 3 organs that most plants have. So roots, shoots, leaves.
Mhm.
All right. Yeah.
Yeah. So like I said, non-vascular plants, so your bryophytes, mosses, hornworts, liverworts, they do not actually have leaves for very pedantic reasons. It is solely found in vascular plants.
Gotcha. Those other plants have leaf-like structures, I assume,
Yeah.
that are doing the photosynthesizing.
Yeah, they are, I would describe them as leaf adjacent.
Sure.
But
Leaf-esque.
for technical reasons, they are not literally leaves.
Right.
Makes sense.
Leaves are the main. I mean, this is where plants get their energy from.
Mhm.
Leaves are photosynthesis. We did a whole episode about photosynthesis. That was episode 145. Extremely important to plants being plants.
Yes.
Leaves got a lot of shout out in that episode because
Yes.
that's where it happens.
Yes. It's the room where it happens, as it were.
That's true. With the broad scale description out of the way, let's go a little deeper. The anatomy of a leaf.
Okay. So we're going to kind of talk about this on 2 different scales. So we're going to talk about it on a cellular scale and then like a macroscopic scale, because those are kind of the 2 important levels that things are happening at. So if we are talking about the cellular structure of a leaf, don't worry, this is going to be real brief. I like to think of it as a sandwich. From the top to the bottom, it is epidermis, palisade layer, spongy mesophyll, epidermis. So the stomata, which are the pores that allow gas exchange and allow transpiration, are typically on the bottom of the leaf. So
Okay.
you have the guard cells that allow the stomata to open and close, and then you have the epidermal like cells themselves. Sometimes you'll have waxes or hairs. So in general, the cuticle, the wax keeps the water from coming in and out when it's not supposed to, protects against water loss, regulates gas exchange, secretes metabolic compounds. But the palisade layer, which is the second layer from the top, is the most exciting. So that's where photosynthesis actually happens, is in the palisade layer. So that's where you have this high concentration of chloroplasts. Beneath that, you have the spongy mesophyll, and that's basically where the gas gets trapped that is incorporated into photosynthesis. So that's where CO2 hangs out. That's where the water vapor that gets incorporated into photosynthesis hangs out. So yeah, basically you have a sandwich, and that is what a leaf is, which is wild given how many different types of leaves. Regardless, they all have pretty similar structures. Like the specifics are different, but by and large, you have epidermis, you have the palisade layer and spongy mesophyll and stomata, like all mixing together. It's actually kind of wild.
Cool.
Does that mean that leaves take in light from above and gas from below?
Correct.
Huh.
Well, and it's neat that the part where that the gas intake and water vapor and stuff is spongy, that it's a leaf lung.
Yes.
Like it's spongy
Yes.
material, like the alveoli of our lungs and everything.
Yeah. Well, when you described the spongy mesophyll as being where the gas gets trapped, my immediate thought was like, all right, cool, I get to make a joke about farts. But then
Yes.
you continued I was like, oh, these are not leaf. This is the opposite.
Yes.
This is the gas that's coming in.
Yes. This is leaf breathing.
Leaf breathe. These are leaf breaths.
Yes.
Which is less funny. Interesting. I guess that is a really. I'm already learning stuff about leaves. This is great.
Well, and it's like, because we know that leaves have an A and a B side, because you can see very clearly that they are different.
Yeah.
Like when a leaf's upside down, you know it's upside down.
Yes.
I just had never put much thought into what jobs those two sides were doing differently.
It makes now that I'm saying it out loud, it makes all the sense in the world that the underside of the leaf is not the side doing photosynthesizing.
I just didn't think about the breathing being specified there.
Surprise!
Cool.
Literally, step one. I'm glad that I'm blowing your minds already.
Biology, it just makes sense.
So don't worry, that's the last time I'm going to go in specifics about like that level of detail. So that's the cellular structure of a leaf. Important to note, I mentioned it in the palisade layer. So you basically only get chloroplasts where places you're doing photosynthesis. So unless your stem is doing photosynthesis, you're pretty much only getting chloroplasts in the leaves, which means that when you learned in a biology class, this is what a plant cell looks like, and you learned about, chloroplasts and whatever, it was a leaf cell.
Yeah.
Like, you were very misled. It was just a leaf. We said it was a plant cell. It was a leaf cell, specifically.
Makes sense.
So that is, broadly speaking, the inside of a leaf. Okay, so at a bigger level, the full leaf level, so there are, generally speaking, I'm so sorry. I'm going to put things in so many categories throughout this episode. This is the first of the categories. So there are simple leaves and there are compound leaves. Simple leaves are, I don't know if it's obvious to say, basically, if you think of a leaf, right? Like, close your eyes, think of a leaf. Or if you envision like the maple leaf that's on a Canadian flag, that is a simple leaf, right? There is only one part to it, Then there are compound leaves. So that's going to be like hickory leaves, or if you think about who else has compound leaves? Hickories, walnuts, things like that. Basically feather-shaped leaves, those are compound leaves.
Oh, okay.
Yeah. So simple leaves have much simpler structures. Unsurprisingly, it is in fact in the name. The nice thing is, genuinely, most people on the planet are familiar with some leaf of some kind. So whatever your favorite leaf is, visualize that, and I'll walk you through the names of the parts. So you have the lamina. So that is the actual leaf blade or leaf frond, the part that's green and, floppy. That's where the photosynthesis is actually happening. Holding that up, you typically, not always, but typically have what's called a petiole. So that is the leaf stalk. So if the leaf is an umbrella, this is the handle of the umbrella.
Yes.
This is the part that you hold while you're fanning somebody with a giant leaf.
Yes, exactly.
Precisely. Yes. So that's the petiole. In order for a leaf to be a leaf, it has to be vascularized. So there has to be tubes running through it. So you have some sort of vasculature. So, and that's your venation. So they can be parallel veins, they can be palmate veins, they can be netted veins, but there has to be some sort of hydraulics running through the leaf. And then very simply, the edge of the leaf blade is called the margin, which makes perfect sense. And then in monocots, you also will have what's called a sheath, which is basically, if you've ever stared at a blade of grass, and I realize that might not be something that most people have done.
That you're a huge dork.
That has been reinforced to me multiple times when preparing for this episode. But for many monocots, you'll have kind of a little bit of extra tissue on the bottom that was basically protecting the leaf as it was growing.
Yeah, because they kind of unroll from that.
Yes, exactly. So that's a simple leaf. Very simple. It's in the name.
Cool.
Compound leaves have pretty much the same things, but more. So the actual stalk at the end of the whole compound leaf, that's also called a petiole. But the little bitty baby stalk on each of the leaflets is called a petiolule.
Petiolule.
Yes.
Okay.
I know.
And petiolule is the next one.
Sure, and then it keeps
petiolule.
I'm very certain that everyone who has ever learned that word has made that joke.
I mean,
I certainly have.
Right? Like, I'm assuming that's the joke that was made when the name was made.
Absolutely.
It's like, well, there's more of them. Okay, well, you know what to do.
Exactly. And then the rachis, which is the same name as the part in the feather, is basically the main stem part of the compound leaf. So you have the petiole at the bottom, and then the middle stalk is the rachis.
Cool.
And then, if you have more than one subdivision, you have the rachilla.
The racules.
Exactly. So, rachis, rachilla.
All right.
And then so the technical name for the divisions is not leaflets. We call them that, but technically they are pinnae. So a pinna is a primary division, and then a pinnule is a secondary division.
Okay.
And I imagine that I wonder if it would continue if you have tertiary divisions, that it would be a pinnule, but I don't actually know. I would call it that.
For the sake of this discussion, we will assume that that's true.
It's canon to this episode.
Absolutely. So broadly speaking, that's it. So you have, and this is true of all vascular plants. Their leaves fall into, even if they seem very different at first, you can begin to break down like, oh, okay, so like if you think about like a pine needle, it's not flattened, but there is an edge to it. So there's definitely a margin. It has a petiole. Well, it doesn't actually have a petiole, but it has to attach somehow. So this is very broad strokes, because there's a lot of things with leaves, yo.
Yes.
And I'm guessing that what makes a compound leaf not just a bunch of leaves is development. Like,
yes,
the way it develops makes it that it is developing from a single leaf part into these multiple structures.
So you have hit on one of the things that I find abundantly interesting. So one of the things that I find interesting is, what is the functional unit of a compound leaf?
Yeah.
So in paleobotany, for logistical reasons, we have to assume that a leaflet is equivalent to a simple leaf.
Yeah.
Because unless you find that leaflet, in situ associated, you don't actually know it's a compound leaf.
Yeah.
But that means then that, like, you're making a lot of assumptions. And so when I was living in Kansas, I spent a spring staring at a lot of compound leaf trees as the leaves were flushing to figure out, what did the different compound leaves look like as they flushed? Did they treat the entire thing as a leaf? Or did, like, if it had a terminal leaflet, did that grow and develop at a different rate than, the lateral leaflets? And of course, being biology, the answer was, it depends.
Yep.
Well, there's a lot of different kinds of leaves out there.
Yes.
Is my understanding of the world.
Yes, this is true.
In fact, you have alluded to the fact that we're discussing this very basic, generalized structure of a leaf, but that leaves are present in all vascular plants. That means all the gymnosperms, episode 155, and all the angiosperms, episode 57, which is all the plants.
All of them.
Excuse you, there are also ferns.
And also ferns,
Yes.
that's true. Which is some of the plants.
Yes, exactly.
What is some of the ways that leaves modify that structure?
Oh, goodness. So, in biology, it feels like things are either way easier than you expect or way harder than you expect. Just in general, I feel like that nothing is actually what you expect it to be. And in general, it's actually really easy most of the time to look at a leaf and know what type of plant it is, like, broadly speaking. If you look at a fern, you can be pretty confident you're looking at a fern, right? If you look at a gymnosperm, mostly you can be pretty confident you're looking at a gymnosperm, especially if you're looking at conifers. Like, those, that's what we think of when we think of gymnosperms.
Those needles.
Exactly. Monocots can be really easily differentiated from dicot angiosperms. So, which is, I think, really fascinating, because, like, obviously there are some groups where there's a lot of overlap and it can be misleading, but in general, they're really easy to differentiate, which seems too easy. But in general, so ferns have fronds. And when I was looking into this, man, some people are very adamant that frond can only refer to ferns.
Gotcha.
Some
All right.
people will also, I am into this group, will also use the word frond to refer to the leaves of palms and cycads.
Yep.
They have the same vibe, right?
Yes.
So
Sure.
that seems frondy to me.
Yeah, I mean, like, when you say frond, one of the first terms that comes to my brain is palm frond.
Yes,
Right.
exactly.
And if not frond, why frond shaped?
Exactly.
You can have that one for free.
I will hold on to that. I will make that joke to my friends tomorrow. So in general, gymnosperms, primarily conifers, tend to have needle leaves, all leaves, or scaled leaves. But it can vary. In particular, the Natalians, so that group of gymnosperms, really like to look like other things. So like Needham and Ginkgo look like dicot angiosperms unless you look real close. Ephedra doesn't even look like it has leaves. My favorite is my dear friend Welwitschia,
Yep.
which only has 2 leaves. And the leaves resemble, if you look at them, they kind of look like monocots. This is a fun fact. When I was looking up Welwitschia, because I went real deep before I realized, okay, back it up. I found out that the Afrikaans name for Welwitschia is Tweeblarkania dude, which means 2 leaves can't die.
There you go.
Just like, yep, correct. That's Welwitschia. So the different groups of vascular plants all have pretty widely varying leaves. I'm going to try to be as fair to all vascular plants as I can, but I will be biased towards angiosperms. That's what I study. They are my true love, and I think they have the best leaves.
Well, I mean, they rule the world, so we have to at least...
Right. They are the
Yes.
current plant overlords.
Yeah, we have to play to them a
Exactly.
little bit.
I, for one, welcome our angiosperm
Yeah.
overlords.
They're right outside the window.
Yeah, they're
I
in
can-
the room with us.
That's true, they are.
They are, actually.
Yeah. I got 2 of them right there.
Within plants, who do you think? No, this is my favorite part. When I ask you questions I know you don't know the answer to.
Oh
Right.
boy.
Who do you think has the largest leaf of any vascular plant? And you can be general. I won't ask you for a species. That's rude.
Oh man, I feel like I knew this once upon a time. Because aren't there those like weird ones that grow on the floor with those big honking leaves?
Yeah.
That's Welwitschia.
Oh, okay, that's Welwitschia.
Yes.
Great. That or palms.
Yeah, the only one that comes to mind are the elephant ear plant, which I don't actually know what
Oh.
kind of plant. I guess that's a monocot because of how it grows and unfurls, but I don't actually know what kind of plant that is.
Let's learn together.
Y'all did great. Okay, so the largest leaf of any vascular plant is a palm.
All right.
Ooh, hey, nice.
So it's one of the raffia palms, and I could not track down this paper. I was so annoyed. I tried so hard, but it has been reported that raffia regalis leaves can be as long as 25 meters long. That's 82 feet, y'all.
That's a leaf the size of a blue whale.
Yeah,
How does it? Where does it put it?
at the top.
Like, where does it put it all?
On
Like,
top of everything else.
Yeah.
Yes, exactly.
that's ridiculous.
So most of them are not that long, but they're still
Sure.
like 30 feet long, which is absurd. So
Wow.
we were talking about other big leaves, though. So the greatest surface area is one of those ones, kind of like what you were talking about, Will. So it's Cocoloba gigantifolia. So there's this GIF of Sir David Attenborough hugging a leaf. So that's one of these. It has the
Yeah.
greatest surface area of any leaf. So they're about 2.5 meters long, so 8 feet long, and about 1.5 meters wide, so like 4.5 feet wide. They're
Nice.
like blanket size.
Yeah, that's a blanket I
Yeah.
could use.
Yeah.
Or just a big old solar panel.
Absolutely. And then the smallest leaves belong to ferns and angiosperms. So there is a zola, which is a very famous fern if you're in paleobotany. They made the Arctic green in the Eocene.
Cool.
So their leaves are about 2 millimeters long.
Aww.
I know, sweet babies. And then lemna is an itty bitty angiosperm, and their leaves are about 4 millimeters long.
Just taking up a photon at a time.
I feel like just a little nibble. Boop. I feel like I looked at lemna in like a biology lab.
Oh, I'm sure you did.
Is this like an aquatic?
Yes, it's duckweed.
That's yeah.
Right.
I spent hours counting lemna leaves under a microscope. It was no offense to lemna or to plants in general. It was not fun.
That's very fair. But like, think about that. You have 82-foot long leaves and 2 millimeter leaves.
Yeah.
Wild. And so finally, my favorite category of talking about leaf diversity is shape.
Mm-hmm.
I am a nerd about leaf shape. I get real excited when I learn about new leaf shapes. I have a whole book about leaf shape over here. So in general, if you're going to get real pedantic, so my book classifies leaves by their apex shape to the top of the leaf, their base shape, the bottom of the leaf, and their symmetry. I will not be getting into that because that is a level of detail that no one but me cares about.
So when we do the spin-off podcast, that'll be one of the episodes of Leave It to Allie.
Absolutely. In general, there within simple leaves, there are dozens of leaf-shaped, lobed and unlobed. I'll go through a few of them because these tend to be very common, especially with trees of dicot angiosperms. So an elliptic leaf is leaf-shaped.
Yeah.
Like, that's it. The widest part is the middle fifth. It tapers at the top and the bottom. Leaf-shaped leaves are elliptic. Then you have your ovate and your obovate. So basically, is the widest part at the top or is the widest part at the bottom? Kind of like egg-shaped or upside-down egg-shaped.
Yep.
Oblong is basically, a lot of it is parallel in the middle, but then it tapers at the top and the bottom, as opposed to be kind of rounded on the sides. And then linear, the length to width ratio has to be at least 10-1. Has to be at least 10 times longer than it is wide.
Okay. Long strap-like leaves.
Yeah.
Exactly. So that's what qualifies as linear. So those are just a sprinkling of possible. I could go so hard, but I'm behaving myself for y'all. Within compound leaves, so your leaflets can have those shapes too, right? So you could have your pinnae could be elliptic, obovate, etc. And then in addition to that, we have words that describe the amount of dissection of the division. So pinnatophyd versus pinnatisect. So this is primarily when we're talking about ferns. So basically, do you see lamina on the sides of the rachis, or is it basically like its naked vein in between? You can also have varying numbers of subdivisions. So you can have a unipinnate or just a pinnate compound leaf. So that's the one that is feather shaped, right? So you have the stalk and then you have leaflets coming off the sides, unipinnate. Bipinnate is when you have the stalk and then each of what was the leaflets on either side has another stalk coming off and then leaflets coming off of that. And then you can have tripinnate where you have one stalk and another stalk coming off and then another stalk, and that's where the leaflets are. And tetrapinnate, it goes like, leaves don't mess around.
Yeah, just keep just fractaling out.
And these are, I think, what you're describing of as very classic fern shape. You have a stalk with a bunch of little leaves sticking off the side, and then sometimes you have a stalk with a bunch of little stalks, and each of those have little leaves off the side.
So this is very common in ferns. It's also very common in legumes, so fabaceae.
Mm-hmm.
The Kentucky coffee tree has way more divisions than you expect until you start looking at it. Like that's a lot of leaf. But yes, so this is very common in ferns. Like they have cornered the market on compound leaves.
Yep. Cool.
So a couple more things you can kind of, when you're classifying shape, there are some special leaves that I will not be getting into in this episode because I have talked about them a whole lot before. Highly modified leaves, such as carnivorous plants.
Yeah.
Yep. Episode 105, carnivorous plants.
Yeah, for whatever reason, pictures just don't really fit into any of the size classes or shape categories I talked about before. So yeah, any sort of modified leaf like that. When we talk about the margin of leaves, we talk about them being toothed or untoothed, and that is very important to me in my research specifically. And then another way we will classify the difference in leaf shape is by looking at the venation. So again, is it palmate venation? Is it net venation? Is it parallel venation? So it's important to note, this is something that has been just beaten into my brain. Leaves are the worst way to identify a plant. They are very plastic in their shape, which we'll talk about more. And so there's not necessarily a relationship between like, this is who, there are many different groups with the same shape leaves. Elliptic leaves, real common. Like leaf-shaped leaves, there's a reason we think that's the leaf shape.
Yeah.
But if you look at the venation, particularly the higher order venation, so the primary venation is the midrib, the secondary venation, is the veins coming off of that. So when you get to third and fourth order, those are actually can be used for identification. And in the fossil record, that's sometimes the only thing we have.
Yeah.
It's funny because so much of your description of leaves up until this point has made me think a lot about teeth and comparing them to the teeth of vertebrate animals. Because, like you said before, as with leaves, you can typically look at a tooth and know what general type of animal you're looking at.
Yep.
Teeth are highly specific and highly specialized between species, especially in groups like mammals or in certain dinosaurs, in part because they are the main method that the animal is retrieving its food. And then you said, leaves are the worst way to identify plants. And I said, that's different.
Yep.
That is not like teeth.
That's not like teeth.
Yeah.
Exactly, because mammals are only teeth in the fossil record.
Yes, exactly.
It is very funny because leaves are the primary way that most people interact with plants, and they're a terrible way to identify them.
Yeah.
So don't feel bad. You were set up for failure.
Yay.
So lots of types of leaves.
Yes, that's it. That's all you needed to know.
Many much leaf.
Yes.
Well, we've been in discussing sort of the diversity of the structure of leaves. We have also been touching on so far throughout this discussion what leaves do. So let's talk a bit more about leaf function, starting, of course, with the primary function of leaves, which, as far as my knowledge goes, is almost universal across leaves. That the thing they do is they collect sunlight.
Correct. You're right. The primary function of leaves is photosynthesis. I did lead with that when I called them the primary photosynthetic organ.
Yes.
But that's it. Like, the reason that plants have leaves is photosynthesis. So just to remind us, I know I've already talked about photosynthesis, so if you want me to get real in the weeds about it, go check that one out.
Episode 145.
But in general, the stomata allow the CO2 to pass through the spongy mesophyll into the palisade layer. The water travels into the leaves via the veins, and then photosynthesis occurs in the palisade layer. That's it. They are very specialized photosynthetic organs. They're very good at their job. Yeah, that's it. That's all there is to say. Leaves are for photosynthesis. Yeah. Everything else is secondary.
We've made this comparison before, and I've heard other people make this comparison, but they really are just little biological solar panels.
Yeah,
I mean, absolutely.
That's 100% what you're dealing with, and it was one of the first thoughts I had when I took botany and like saw the structure, and it was like, yeah, those are just little photocells.
Yep.
Oh yeah. Well, the funny thing about plants, and I could kind of mention this anywhere, is that they're modular. Right? Like, there are 3 types of organs in a plant. That's pretty much it. There are some things and more specialized things, but in general, you have roots, you have shoots, you have leaves. And that means that you can kind of build on them in ways that you can't with animals. And it's just so cool. Like, and cell walls are fun too. Like, that opens up a whole world of possibilities.
Well, like you were saying with the compound leaves, that you can just keep branching off.
Yes.
And just branch off of a branch off and branch off of a branch. Like, you can legitimately just redo, just Do it again. Do it again.
Copy paste.
Yep.
So leaves, first and foremost, photosynthesis, collecting sunlight. However,
Yes.
like so many organs in biology, and a fact that I think is obvious to anyone who has listened to our other plant episodes or who has ever heard of plants,
Yes.
leaves do other stuff.
Yes.
They have been modified and repurposed for other functions. What are some of the other things that leaves do?
So this is closely related to photosynthesis. It's friends with photosynthesis, but it is not photosynthesis. So transpiration, it happens through the leaves. So transpiration is the movement of water through the plant and evaporation. So it's a passive process. So basically water comes up through the roots, goes through the shoot, goes out the leaves. So the movement of water is important for photosynthesis, but actually releasing the water into the alphabet? No, atmosphere. Releasing the water into the atmosphere is a passive process that happens in the leaves. So it is controlled, the process is controlled by concentration gradients. So basically, it is wetter inside the plant and drier outside of the plant. And so things move from wet to dry.
Yep.
That's it. My favorite thing, and I think I've mentioned it on here before, but I'm obsessed with this fact. The reason that water moves faster than CO2, so water moves out of a plant faster than CO2 can move into a plant, is because one, the concentration gradient is much steeper. It's much wetter in a plant and drier in the atmosphere than the difference between CO2 in and out of a plant. But also, CO2 is physically a larger molecule than water, which is just wild to me. I will tell everyone this because it's one of my favorite fun facts, and it makes everything make sense. But when we talk about evapotranspiration, we're always talking about efficiency, and it comes across as a bad thing. Like, oh no, the plant is losing water. Oh no, it's going to dry out. And yes, too much transpiration, too much evaporation, it is bad. When I lived in Kansas, it was really, really dry there. There was very low humidity, and my plants were not happy. It was just sucking the water right out of them. But it could be a good thing. So it actually cools the plants.
Yeah, like sweating.
Exactly. So it allows the plants to, not overheat, because there are temperatures. If the plant gets too hot, it can't photosynthesize.
Yeah.
So you gotta keep cool. And it changes the osmotic pressure of cells. So the thing about plants is they have cell walls. So if you have too much water inside, it can't really go anywhere.
Yeah.
And that's bad.
Yep. Then your cell explodes.
Yeah, we don't like that. But if you have too little water in your cells, the plant just wilts. That's what wilting is.
Yep.
Because they have turgor pressure. They have water holding them up.
And that's why they can bounce back after wilting once you give them water.
Exactly. Plants are the best. And this also enables the flow of nutrients through the plant. So in addition to that, my favorite, bar none, thing that leaves can randomly do, I've had multiple conversations with people about this week because I can't stop talking about it, is guttation. Are you familiar with guttation?
Guttation. Guttation.
T-T-A-T-I-O-N? Yep.
Guttation. Nope.
Okay.
That was a guess. I don't know.
Yeah.
I'm not at all familiar with it.
You sounded so confident. Oh, yes, I can spell this word.
I know a Steve guttation.
So guttation is not dew. So if you see a plant that has little drops of water on each of the tips of the leaves, this is very common in strawberries, it is not dew that has condensed on the plant. There are drops of sap that have been exuded from the plant margin of some vascular plants.
That's real plant sweat.
Yes. So the way that this works is it's most common at the leaf apex or leaf teeth. So like I said, it's very famous in strawberries. You if you look this up, you'll find lots of pictures of strawberries. But because I'm a nerd about this, I look for it in different plants. So vitis, so grapes do this. My spider plant does it. I've seen it on hostas. So Basically the water is coming out through these specialized cells called hydothodes. So this is most common at night when the stomata are closed, right? So there's no transpiration happening. So they're not able to release water to the atmosphere. And if the soil moisture is high. So I always describe it as their feet are wet and they want to fix it.
Yes.
So basically the water enters through the roots and the root pressure forces water up and out of the plant through these specialized pores.
Cool. It's just an alternative exit.
Yes.
To get rid of extra water.
Yes. And the cool thing about the hydothodes is like basically it's a pore, it lets the water out because otherwise the water has no place to go. And so it'll basically pool in the cells along the leaf margin. And then that's bad because again, you have too much water in your cells.
Yeah.
I am obsessed with guttation. I think it is so cool. Fungi do it, but what they do is completely different. It just has the same name. But yes, guttation is one of my favorite random things that leaves can do.
Is that pooling in the leaf why overwatering your plants can cause the leaf edges to go brown and stuff?
Yes.
Oh.
It didn't have a place to go.
Yeah. I was always kind of confused by how overwatering was killing the leaf.
Mm-hmm.
Like, I make sense like root rot and fungus and all that stuff. That makes perfect sense. How it was killing the leaves was baffling me. It's drowning the edge of the leaf.
Plants are tubes.
Yeah.
That's it. They didn't have anywhere to go at the end of the tube.
Yeah.
Cool.
Well, it's odd to think about drowning a part of the body of the plant.
Yes.
Yes.
And not, from our perspective, drown, you drown.
Your whole body
That's
does it together.
The body,
Yes.
we're all in this as one. So that's interesting. So it's just the cells are just dying.
Yeah.
Along the outside margin of that leaf.
Yeah.
Interesting. So when we see dew dripping off of a plant, I assume that dew also tends to gather and drip off the pointy parts of the leaf because of the way that water flows.
Correct. So yes, because it's the difference between the water is coming up from, the soil to be exuded out of the plant versus from the atmosphere that is condensing on the high points of the plant.
Right.
So it's very funny. I think about this a lot. So I was actually thinking about this. It's been really wet here recently, which means that because there's high moisture in the soil, the plants are trying to get rid of the extra moisture. And so I was looking at these grape leaves, and it's fascinating because you can see both the condensation and the guttation.
Yeah.
So the condensation is on the surface of the leaf. It's little spots on the surface of the leaf. And then the guttation is only on the teeth.
Yeah.
Gotcha.
It's so
very cool. That is very cool.
I think
Neat.
it's so pretty. I have taken so many pictures of leaves undergoing guttation. So in addition to that, I'm going to kind of briefly cover a bunch of other ways that leaves can be modified. I am so sorry. I can't go over all of them. I have taken out so many things because I realized okay, that's too much. We got to back off a little bit. So basically, this is a whistle-stop tour of some other ways that other uses for leaves. So basically, ways that leaves can be modified. So I'm going to start with the ones I've talked about in different episodes, and then talk about ones that I haven't covered before. So, like I said, carnivorous plants, they have modified their leaves.
A bit.
Just a little bit.
Just a smidge.
There are also many plant defenses that are derived from leaves. So either they can be secondary metabolites, right? So these are compounds that the leaf is making. So if somebody chews on it, it ruins their day. But it could also be physical structures that are either derived from the leaves themselves or modified leaves.
Mm-hmm.
So those are ones I've covered before.
175 episode about plant defenses, where we talked about those secondary metabolites, and then physical defenses we talked about a whole bunch in episode 185 about cacti.
Yes.
Which is the example that always comes to my mind first when I think of highly modified leaves.
Yes.
The needles on a cactus.
Yes.
Those are highly modified leaves.
Yes, exactly. There are so many other things. So kind of talking about plant insect interactions a little bit. There are a couple different categories of things that plants will either do to their leaves or do on their leaves. So extrafloral nectaries and food bodies.
We just recently talked about a news that was a study about extrafloral nectaries.
I loved extrafloral nectaries. So simply put, they are nectar that aren't in the flower. That's all. So extrafloral nectaries are often on the leaves. So like associated in like the leaf pits, like I don't know what I would consider the armpit of, like
Right,
between
yep.
the petiole and the blade.
No, it's as soon as you said leaf pit, I was like, yeah, no, I know
absolutely.
which part you're talking
Yeah,
about.
that's where it is. So those are ways that plants will tempt insects or food bodies. So these are different ways that, different little treats. There are so many types of little treats that that plants will make for their insect friends to entice them to stay.
Yeah. We've talked about plants do a lot of things to attract insects for
Yes.
purposes like pollination and whatnot. In the news not too long ago, we talked about a study on plant nectaries that are used to attract ants, to incentivize the ants to treat this plant as an important resource and therefore defend it.
Yeah.
Instead of developing its own defenses, it hires some ant bodyguards to defend the plant.
Perfect transition. And another thing, in addition to feeding the ants, they'll make them a home.
Yep.
Yes.
Domatia.
Oh, hey, I got it.
Yeah. It literally means house.
Yeah.
So, which I love so much. So ant domatia, or the Myrmecodomatia. There we go. Ant houses. That's what that means. Those tend to be real big. So if you look at like acacias, they will have them, unsurprisingly.
Yeah.
They're famous for their relationship. But those are often modified leaves.
Gotcha.
Or Acarodomatia, which are mite houses.
Sure.
Those are often, I'm so sorry to make this comparison, but someone made it first and I can't unsee it. They are the hairy armpits on leaves. So basically, there's a bunch of trichomes, which are leaf hairs, that are really dense. And they tend to be kind of where multiple veins will come together on an angiosperm leaf. So it'll be very dense, and the mites can hide in there, which is really neat. You can see that in the fossil record, actually.
Ooh.
But we have people at Michigan who are working in our herbarium who are looking at our herbarium sheets to see if we can find mites in the domatia on the herbarium sheets, so that we can do basically a survey of what is the mite diversity, and are they generalists? Are they specialists? Are there species that are only found in certain species of plants? Because a lot of temperate angiosperms actually have domatia, little mite domatia. It's very cute.
Neat.
So in addition to the plant and insect interactions, there are just a lot of structures that plants make out of their leaves. So bulbs are actually derived from leaves. So like tulip bulbs.
Yeah.
Those are leaves. Which makes sense if you've ever like peeled one apart.
Yeah.
Like, oh yeah, those are leaves.
Huh.
I know, right? So tendrils of some climbing species are just modified leaves.
All right.
That they're using as grappling hooks. Some of them will twine around. There are some species that literally use like grappling hooks, right? They have little spikes coming out. I love them so much. Sometimes plants will make leaves out of leaf material that's not the blade.
Oh.
This is so weird to me. So phyllodes are a photosynthetic organ that is leaf-shaped, but it is derived from the petiole.
Yeah.
So it is not the leaf blade, it is the petiole, so the leaf stalk, that is modified into being photosynthetic. It is wild.
Yeah, why?
Because it's there.
Yeah.
Is there also a leaf blade?
Sometimes. Yes. So I was learning. Okay. So I asked my colleague, I was like, hey, what's your favorite fun fact about leaves? And instead of giving me an answer, he gave me a book.
This is what happens when you talk to nerds.
Yes, exactly. And so I was looking through the chapter, or like the couple pages on phyllodes, and in some species of Fabaceae, so legumes, so acacia and friends, the juvenile leaf looks like what you would expect an acacia kind of leaf to look like. It would be bipinnate, right? So you got the stalk, you got little leaflets coming off of it, real cool. as the leaf grows, or as the plant matures and you have more mature leaves, the stalk, the petiole, will begin to develop into a phyllodes. So you have what looks like a very standard simple leaf, and then coming off the top of it is a compound leaf. Until eventually you only have the phyllodes, but you can have both.
Weird. I didn't think of it coming off the end of the leaf.
Yeah, because it's a stalk that is pretending to be a leaf. You have the leaf on top of it.
This is more leaf in your leaf.
Yeah.
Yes, exactly. So this is all actually found in both euphorbs and cacti.
Huh.
Yeah. Weird,
That
right?
is very odd.
They're so strange.
I know I've seen leaf stalks before that have like flattened parts, but not quite to that degree. That's
well, there are sometimes there are wings.
Yeah.
So the phyllodes is different than the wing, because the wing is just laminar material that extends onto the petiole or onto the rachis. I know.
Sure. So just the regular leaf blade
Yes.
shifted to another place. This is the actual stalk spreading out and flattening out.
Yes, doing its best leaf impression. It's wild.
That's cool.
And then finally, leaves are actually very important for reproduction. So in flowering plants, petals and sepals, so which are basically the layer of a flower that is before the petals, so like outside the petals, those are modified leaves.
Makes sense.
Right? Not surprising. And then in some plants, you also have bracts. So that can be associated with fruits, that can be associated with flowers. So if you've ever seen a flowering dogwood, the white showy petals, those are actually bracts, which are a different kind of modified leaf, different than a petal. So they're just like, we have the stuff. We have the building blocks. Let's see what we can do with it. They're basically Legos.
Cool.
In a decent number of plant groups, so pretty much most ferns and some weirdo angiosperms, the means for reproduction are actually on the leaves. So fern sori, so those are the things that release the spores, will be on the bottom of some fern leaves. And then there are some species of angiosperms where the plant will grow flowers on the leaves, which doesn't feel allowed.
That seems a weird way to do that.
Yep, they decide, and sometimes sometimes the flowers are coming off the petiole. Sometimes it's coming off like the mid vein of the leaf. It's very confusing. I love it so much.
It's just like plating it up on a leaf.
Yes, exactly. Yes, exactly. And then finally, you'll have things like, it's a succulent plant. The common name for it is often mother of thousands. So it'll vegetatively propagate little plantlets on the margin of the leaf. And then they just drop down.
Mothership.
Yes, that's mother of thousands. Yeah. So
also very cool name.
Yeah,
it's a good name. So yeah, basically, I originally again had way more things in this section.
Sure.
I will let you explore, or maybe you can request it. But again, there are not very many parts to a plant, and so they could just go wild. They have the building blocks. It's like, oh, let's try this. Oh, let's try this. Oh, let's try this. And it's really intimidating because it's kind of wild how many things are just secretly leaves.
Yep.
Well, and it seems like of those parts, right, roots, shoots, leaves, the leaves also feel like the part that is most free to be modified and adjusted and take on different shapes.
Yes.
Whereas like roots and shoots, it feels like there's a little bit more of a restriction in how that thing can be shaped, how it can function. Leaves are just kind of hanging there.
Yeah, literally.
As long as you can photosynthesize most of the time, there's room for other functions to pop up.
Yeah,
right.
Like you've got tons of the leaves, so even if you are super modifying them to the point that they can't do stuff, you can still have others just being leaves.
Yes.
Yes, exactly. This is why leaves are my favorite part of the plant, because there's so many ways you can be a leaf.
Very, very awesome.
Aquatic plants will float with their leaves, right?
They can, yeah.
Cool. And then are bromeliads like collecting the little pools of water within the leaves?
They can.
Okay. I'm thinking of other
Yeah.
leaf functions.
Yeah.
Though I know we're not going to do the whole list, but those were other ones that came to my mind.
Yeah. Though it's wild. It's so cool. And just like the different ways, again, I'll mention this real briefly because we're talking about all the weird ways that you can be a leaf. I will only mention this briefly. It's so weird. So my colleague who gave me the book on leaves, so he studies the family Podostomacea, which I don't expect anyone listening to have heard of.
Sure,
I had not heard of it. Obviously, Podostomacea.
I heard of it just a little bit ago.
They're often called riverweeds. They're so weird, like to the point where some botanists think that they are a different group of angiosperms, monocots, dicots, and Podostomacea.
Whoa.
Yeah, okay, they're wild. And so he's been writing this paper, reviewing this group and this family. And it is not always clear what plant organ you're looking at, which is impressive because there are only 3. Right. And so there are some things that may be roots, but may be leaves.
Oh, weird.
I know. It's so confusing. Like, I've seen them. They don't look like most people don't. Like, most people, if you looked at it, you wouldn't even think it was an angiosperm. They are often mistaken for algae.
Weird. Huh.
Anyway, that is a whole digression that I could get into.
Put in your requests now for an episode about riverweeds.
Yep.
Yes.
So leaves, highly variable.
The best.
This is an episode where we are just scratching the surface of the incredible diversity that is leaves. I'm going to ask you a question, Allie. This can be a real quick one because it is a question that is not on the outline, but it occurs to me that some people might be wondering because it is maybe the most common leaf question.
Okay.
Why do leaves change color in the fall in temperate zones like where we are?
Okay. Very brief, because I could go so hard on this. So that is related to the leaf economic spectrum. So basically, the amount of energy put into a leaf versus how much energy you get out of a leaf, right? So that is why you have deciduosity. So that's why some things are deciduous, is because there is a period of time when you're not getting anything out of the leaf, so you're not going to put a whole lot of things into the leaf. The reason that they change colors is because the plant is like, I am not just dropping these nutrients on the ground. And so they reabsorb as many as they can. And then when they drop the leaves, they then can reincorporate the rest of them through the leaf litter.
Nice.
Yes. Plants are very efficient. I love them.
Well, fantastic. Lots of great leaf stuff. We will have Allie share with us a bunch of links and photos so that this diversity of leaves we have talked about, you can also see some of and navigate to in the blog post. I think this is a great time for us to take a short break. And then after we get back from the break, we can talk about the past and start getting into the questions of leaf evolution and the fossil record.
Yeah.
Where Allie will be even more excited.
It's true.
All right, Allie, tell us, if you would, about the origins, evolutionarily, of leaves.
All right. This is one of my favorite times when I get to ask you a question that I know you don't know the answer to, which is my favorite kind of question. Okay. How many times do you think leaves evolved?
Yeah, this is in the outline where I put this question here to ask how many times leaves evolved. And I just reread it and went, oh, yeah, that's an interesting question.
I don't know.
I'm going to guess that I'm going to say one of two answers.
Okay.
Either one time or 100 times.
Yep.
Wow. Okay. Yep. It's between 1 and 100. You are correct.
All right. I was the closest without going over.
Objectively. True. Okay. So, all right. But I'm not going to tell you the answer just yet. We have to go on a journey first. Okay. All right. So around 430 million years ago, the innovation of shoots and leaves. So originally you had just shoots, and then the second plant organ was roots, and then you have leaves. So the innovation of shoots giving rise to leaves underpinned an explosive radiation of vascular plant form analogous to the Cambrian explosion of animals.
Makes sense.
That makes sense. We've talked about this. The common example that we talk about with an explosive radiation of forms is flight.
Mm-hmm.
Yes.
We've talked about every time animals have evolved, all 4 times that animals have evolved flight, they immediately become extraordinary, diverse, and widespread.
Yes.
Because you did a thing that has opened up an entirely new lifestyle.
No, you gained a superpower.
You gained a superpower.
Yes.
In flying, it makes perfect sense that we talked about in the trees episode 73, that when plants evolved wood.
Yep.
Suddenly forests because you gained a superpower.
Yes.
It makes perfect sense that when you evolved solar panels.
Yes.
Then now you get to just do solar panels as much as you want.
Exactly.
Boy, are we behind on that trend.
Right?
I want my solar
There were a
panels.
lot of Silurian leaves who were like, I don't think that's the way to go.
It is, well, I'll talk about this a little bit. It is funny how many like early land plants got real close to leaves, but like, nah, we're not going to go the whole way, which I found very funny. So evolutionary changes to shoot morphology is what led to the origins of leaves.
Sure. Outgrowths of the stems.
Correct.
Essentially.
Yes, and I'll get into specifically how, but there are polyphyletic origins of leaves.
Dude, that doesn't surprise me.
I know, shocking,
Multiple
right?
origins.
Multiple origins. So we'll talk about that. So this is this is a direct quote that I found very funny. Current understanding of plant phylogeny and leaf development fails to shed light on the origin of microphylls and supports several independent origin of megaphylls. So you're probably like, Allie, I don't know what those things are.
I mean, we do, obviously, but the listeners.
Sure, for the benefit of everybody. I know what micro and mega and fill all mean. So
Correct.
I know
Phil.
They are incredibly misleading terms. So they can either refer to categories of types of leaves or size categories of leaves.
Sure.
Cool.
Which is really unhelpful for Googling things, let me tell you.
Big and or this shape.
Yep.
And
Yes.
small and or this shape.
Exactly.
Hopefully those Venn diagrams never overlap and you're fine.
The big ones don't echolocate. And the little ones do.
Exactly. So
Episode 59 if you want to understand that ridiculous joke.
I thought it was excellent. So in general, there are two categories of types of leaves, and I've only really talked about one of them. So there are microphyll leaves and megaphyll leaves, and the difference is not the size, which is annoying. Whoever came up with these names, I just want to talk.
Yep.
So the first leaves are microphyll leaves. So they are often called lycophyll leaves because they are associated with lycophytes. So they are very common in the fossil record. They are generally small, but they definitely don't have to be. And they are rare in modern plants. So they only evolved once. We're real confident about this one. There is only one origin for microphyll leaves. So today, only club mosses, which are your lycophytes, and horsetails, so equisetum, have them. But it is primary in lycophytes. It is secondarily evolved in horsetails. So they're actually megaphyll leaves that are pretending to be microphyll leaves. Because, of course, this can't be easy. So microphyll leaves evolved from enations on the shoot. Enation is probably not a term that y'all are familiar with. Basically, it's a nubbin.
Okay. All right.
Yep. So you had a nubbin on the shoot that eventually developed vascularization. So that is the difference. Like when I was trying to figure out what the earliest microphyll leaf was, there were so many that got close, but they weren't vascularized. So they had stomata. They were functioning as like a foliar unit. They were functioning as leaves, but they did not have the vascularization that was required. So they were not technically leaves. Oh, it's so pedantic.
But also like weird. How are you doing all the jobs the leaf would normally do?
That's what the shoots were for.
Okay.
Yes. So if you look at the earliest land plants, they look like I'm going to give you a comparison that isn't going to be helpful, but then I'll describe it. Don't worry. Okay, so they look like Psilotum. They look like whisk ferns, which is to say they look like twigs with little balls on them.
Mm-hmm.
Mm-hmm. Mm-hmm.
That's it. That's what they look like. And then eventually you got these little nubbins that formed. There are many, many, many more steps to this that I am not going to go over because even I do not find it particularly interesting. But yeah, basically you have these little nubbins that eventually vascularize, and that's what are your microphyll leaves. So the first one, the oldest known microphyll leaf, is this is a name that I had never said out loud before today, and I have been practicing. We'll see if I can do it. Baragwanthia. So this is a type of lycophyte or club moss, and it is found all over. So Canada, China, Czechia, but it is most commonly found. It was first found in Australia.
Okay.
So most of the record of this is from Australia. And so they are late Silurian, early Devonian, roughly 422 million years. So those are the first ones. There were a lot of things that were quite similar, like Asteroxylon got so close.
A lot of leaf-like things.
Yes.
Which makes a lot of sense.
Yes.
Yeah.
Because leaf seems to be the way to go.
Mm-hmm.
If you want to collect, in much the same way that a lot of animals have developed big, large, broad, flat surfaces like
Yes.
ears or sails or whatever for
Yes.
exchanging heat or for display. Yeah, that shape of thing is going to do the job. If you want to collect air and sunlight,
Yes.
having a big flat thing with a whole lot of surface area is the way to do it.
Yes.
Exactly.
Just physics-wise.
Right, exactly. And that's the kind of I took a class during my PhD. It's the hardest class I've ever taken. It's one of the coolest classes I've ever taken. It was called Ecological Biophysics.
Mm-hmm. Mm-hmm.
And it was literally like, let's do math to figure out how these biological processes work. Oh, it was terrifying, but it has fundamentally changed the way I think about things. And like plants in particular lend themselves to thinking about them from a physics standpoint because they don't move, and that makes it easier.
Well, it's what you were talking about with the way they move water.
Exactly.
They're not pumping it, the way we pump blood.
Right.
It's just capillary action and the shape of the inside of the plant
and
moves water.
And concentration gradients, right?
Yeah.
It's basically just chemistry.
So it's purely the physics of the structure of the plant.
Exactly.
And that's why it makes so much sense for leaves to have that classic leaf shape.
Mm-hmm.
We've talked about this a bunch in episodes like 144 and 179. Surface areas where exchange is happening.
Yes.
We talk about this with animals. We talk about this a lot in regards to body temperature, right? The more surface area there is relative to the volume, that's going to change how much exchange you're able to have. A leaf's entire job is exchange with the surrounding environment.
And a sheet of paper is effectively nothing but surface area.
All surface area. That's all that it has to be.
Your thickness is minuscule.
Just enough space in between to have the layers to do the photosynthesizing and the gas exchange. But other than that, it's just 100% surface area.
Yep.
Exactly.
For physics reasons.
Physics. So they're called microphyll leaves, which is a terrible name. It has to do with the type of vasculature that's going on. They are not always small. So a prime example of this is one of my favorite microphyll plants, Lepidodendron, the scale tree.
Yeah.
Very famous plant.
I love Lepidodendron. They're so cute. They had microphyll leaves that were 78 centimeters, so 31 inches long.
Mm-hmm.
That's not micro. Just saying. So we have established that microphyll leaves have a single origin. Megaphyll leaves do not.
So the mega is the amount of origins. Yeah.
I said 1 in 100, and so far I've been right one time. This is the second one.
Yes. So there are, multiple. So there are multiple origins. So during the later Devonian, in the early Carboniferous, is when you have laminate leaves. So flat leaves with layers, they are laminated, containing multiple veins. So again, this vasculature is really important, differentiating them. So it involved in seed plants, progymnosperms, ferns, and sphenopsids, so like equisetum. But don't think that those are the only types. Don't worry. So those groups all have megaphyll leaves. Now the sphenopsids don't. So equisetum went back and they're like, no, megaphill's the way to go. But in general, so you got your ferns, your seed plants, your progymnosperms, they're generally megaphyll leaves are bigger than microphyll in general. And they tend to contain more than one vein, which is very important. So all modern vascular plant groups, with the exception of the lycophytes and the pretending sphenopsids, the equisetum, all have megaphyll leaves. It has evolved at least 6 times.
Oof.
All right.
I saw estimates between 6 and 9. That is a lot.
Yeah.
So it's fascinating because like even within like sister groups, they didn't get it and then I'll keep it. No, they all got it separately, which is fascinating. It's just why plants. So in general, the evolution of megaphyll leaves has the same steps, which is fascinating. So, you know, in general, there is one way to get megaphyll leaves, and it has been done multiple times, which is not true of things like flight. Think there are slightly different ways to like get at it, but in general, everybody was like, you know what? Yeah, this is how you make a leaf, which is wild. So in general, you need to be branching. Cool. You've got branches. Go team. After that is overtopping. I have no clue why it is called this. It makes no sense to me. But what it means is you have unequal branching.
Well, it's over the top.
Exactly. So you have one dominant stem that is bearing branching systems. Okay. And then you have what's called planation. You make it flat. So everything is in a single plane. All of the branches are in a single plane.
Gotcha.
And then finally, webbing. Lamina forms between the branches on this single plane. So that is how you make a megaphyll leaf. Okay. And it has happened at least 6 times.
Fascinating.
Huh.
So often we think of complex structures like that as originating one time and then everything else just inherited it. Or you get a very similar structure in a bunch of different ways.
Right.
Yes.
Like we've seen with other stuff. The idea that it's just the same process has happened several times is very unusual.
Yeah.
Right.
That's just the most likely way to go about making that kind of leaf.
Mm-hmm.
Those events are going to happen in that order if you're going to end up with a leaf like that.
Yes. You can't have half a leaf, apparently.
Yeah.
Yeah. No, it's really interesting. It's also, there is still so much research and so much debate about, because it used to be thought that if you're looking just morphologically, superficially, morphologically, you have the microphyll and the megaphyll. That's it. There are these, you know, the most modern plants have one, then you have the other one that like, but that's it. There were 2. And then the more you look into it, you realize that the type of vascularization is different in all of these groups. So like, you know, just the way that they're moving, the way the tubes are shaped is different for all of these groups. And so the more you look into it, it's like, oh, this is just there is no single character that categorizes everything, because normally it's like, oh, the leaf gap. So basically there's a gap between the shoot and the leaf in megaphylls, but that's also not really a good character. So when I was trying to dig into this, I had to be very aware of the age of the resource that I was using, like more like, because this has just changed so much, which is fascinating that we, like the paper that I was quoting that was talking about our current understanding of plant phylogeny, like we don't know.
Yeah.
We know it happened multiple times, but we really aren't confident, like, you know, how many times and which groups did it together, and it's wild.
I often wonder when we hit the really tough parts of like plant evolution and understanding the non-animal groups, how much of it is born from us just stuck with an animal brain trying to understand this almost alien form of life compared to us.
Absolutely. I had a conversation with one of my colleagues about this earlier this week, because basically the thing about humans is we are mammals, right? We are animals. And so we really like to try to fit everything into the animal shaped box because it is the box that we understand. And plants are not animals. So, like, which sounds silly, but it is really important to remember. So, even just like basic things that we take for granted, you know, I know lots of people who study animals who are very uncomfortable with studying plants because plants are real casual about whole genome duplication events.
Yeah.
Mm-hmm.
Yep.
Like, that's just a thing you can do with plants.
Plants will also just grow into a different shaped body.
Yeah.
Yeah.
In a different situation.
Yes.
Yeah.
Where a plant can go, you know what? I'm going to be a vine.
Yeah.
Yeah.
Instead of being a shrub.
Like
Yes.
what would typically be a whole different group of life.
Mm-hmm.
And we've been doing it this episode earlier. You know, I compared the leaf to the plant lung. We talked about plant sweat, and like it's hard not to want to compare it to that, but it's like, but that's not actually correct.
Right.
And if you're not careful with it, it will confuse you more than it will help you.
Yes.
Because plants are so very different.
Yes. It's a different kingdom.
Yes.
So you talked about we've got convergence in the origins of leaves. That this is something that has happened multiple times, which is very interesting to know. Are there any other convergences or interesting trends across plant evolution, leaf evolution specifically, that are worth noting here?
I love them all, but I will behave. So it's very funny, right? We were just talking about, man, plants are, you got to keep in mind that plants are different. Plants are so complicated. They can do all these things. I found this quote, this concept that I had never considered before that talks about how much easier plants are to study than animals. And I was so refreshed by that perspective because it's not normally what you hear. So one of the cool things about plants? So studying functional morphology in plants is way easier than doing it in animals.
Yeah.
And it's for two main reasons. One, plants have cell walls.
Mm-hmm.
So that increases their preservation potential. And it minimizes ambiguity of ontogeny. Because you don't have the same sort of like cell death that is more common in animals through growth.
Right.
So that limitation of cell walls makes them, again, they are easier to preserve because they're a little bit sturdier. And they're building blocks. They're Legos. They're not balloons. They are Legos.
Yes.
And then in general, and this blew my mind because I hadn't really, it's something that I talk about. It's you say the words, but you don't think about what they mean. All plants generally live the same way.
Mm-hmm.
They are all photoautotrophs. So, with the exception of parasites, they are all photoautotrophs. They're all doing the same thing.
Yep.
That is not true with animals.
No.
Yes.
There's tons of different lifestyles and metabolic patterns and ways of getting nutrition and such in animals.
And in plants, they're photoautotrophs. They are photosynthesizing.
Yeah, I guess that is a really good point. You find a plant, you know what it's doing.
Yes.
Because it's a plant. Plants do one thing.
Like how it's trying to get to the sunlight or how it's getting its water might be different.
Sure.
But the same ingredients and the same goal.
The parts are going to be there.
Is present every time.
Exactly. They are doing one thing. And that means that the combination of we can see the morphology and there is only one function makes this equation so much easier to balance.
Yeah.
And you know what? That's thrilling to me because I don't normally get to do things the easy way because I study plants. But here's a quote that I found that I really really enjoyed. This uniformity of life strategy in conjunction with developmental constraints also increases the likelihood of evolutionary convergence.
Yeah.
That makes sense.
Yeah, no, absolutely.
It's that modular. Well, they can only do so much. There's a lot of limitations on the evolutionary abilities of plants because you can only modify things so far before either you're outside the bounds of your relatively limited structural basics or you can't photosynthesize anymore.
Yes.
And that you can't have that.
No, there's only one thing you got to do, and it's that.
And it makes sense also from just like the numbers game of if basically all plants are doing the same job, you're going to come to the same answer far more often than most other groups. It's like fast swimming things are all basically shaped the same way because you can only push through water efficiently with so many shapes.
Mm-hmm. Exactly.
And but it's like if everything was a fast swimming animal.
Yes.
If all animals were fast swimming animals.
Yes,
then
exactly.
That's what plants are dealing. So of course you get this ridiculous level of convergence. That's awesome.
I love them. I love plants so much. So there are some trends that we see. It's fascinating. So Paleozoic leaves tend to follow similar trajectories in pretty much every group. So they start out simple and then they become really diverse, right? That's pretty much it. Then they absolutely explode in the Carboniferous. And it's fascinating because, again, like there are some broad trends that you see, but in general, plants are like, I can now do anything. I genuinely just enjoy looking at the diversity of leaves because there are so many ways to be a leaf that just don't feel allowed. Especially because of the research that I do, and I will be talking about that in a little bit. I focus on dicot angiosperms, which is one little corner. I mean, one big corner, but one little corner of the plant family tree. And if you look at everybody else, they're doing way weirder things.
Yeah.
Man, like monocots. How do you even live like that? But so one of the things that I have kind of alluded to, and we'll definitely be talking about more in a little bit, is one of my favorite things that leaves do. It's the concept of phenotypic plasticity. So phenotype is how something looks. The genotype is the actual, the genes. So the difference between like, I have green eyes in my genes and the actual shade of green eye. This is really important to paleobotanical research. So the thing that makes the thing that makes leaves terrible for identification is phenotypic plasticity, among other things. But that's what makes them fantastic environmental indicators. So there are global trends in angiosperm, particularly leaf physiognomy, so size, shape, etc. That is so basically you can see trends in leaf shape on 3 different scales. So there is the evolutionary time scale that makes species. So you have enough differentiation in leaf shape that you have different species. There is the differentiation of leaf shape that happens on a plant lifespan. So this is what the DNA says, but this is what the environment is. So I will make leaves that reflect the environment that I live in. And that can happen on a growing season timescale. Leaves can make, I know, wild, right? So one of the papers that I published during my dissertation was looking at 4 species of grapes that were grown in New York State from 2 consecutive growing seasons, and basically looking at the differences in the leaves along the vines between the 2 growing seasons, and they were predictably different.
That's
because
cool.
We looked at the temperature precipitation from the 2 different growing seasons, and we were like, yes, this is what we would expect to see. These little tiny changes, but predictable changes in leaf shape.
Wow.
Right? It's so cool.
Yeah, that's got kind of the same feeling as like animals that have the winter and summer coats.
Yes.
But this is a different shape of your body, quote unquote.
Right. Well, it's because they are trying to maximize, their because leaves are one of the main ways that plants interact with their environment, right? They're trying to photosynthesize, and they are trying not to dry out.
Yep.
And so that's where this little bit of wiggle room really comes in handy. So when we look at these general trends in leaf physiognomy, there are a couple things that we see. So in general, you tend to have a higher proportion of toothed leaves, so taxa with toothed leaves, in colder environments, which makes sense if you think the Canadian flag, maples have teeth, Canada, cold place. And then you tend to have fewer leaves with teeth in warmer environments. And then you tend to have much larger leaves in places with more moisture, because you don't have to worry about losing water to the atmosphere, because you're still getting it up through the roots. And then places with very low precipitation, you tend to have smaller leaves. I mean, cacti reduce twist their leaves so much they have spines
Yep.
to really like protect the water. But there's also other things, like in general, in cold environments, so environments with the lower mean annual temperature, you tend to have rounder leaves. And in warmer environments, you tend to have more linear leaves. Right, exactly. And this is probably that like surface area thing, right? So cooling. Do you need to keep the heat in? Do you need to dispel the heat? So the relationship between leaf size and precipitation is straightforward but indirect. So basically, the size of the leaf is related to precipitation, but it is not the precipitation that's raining in the leaf. It has to go up through the roots, and so it's not a direct relationship, but it is plant available water. Whenever I give a talk about the relationship between leaf margin and temperature, someone always asks me, they're like, oh, I must have missed it. So what's the relationship between leaf teeth and temperature? And I have to admit, we don't know.
Yeah.
we know there is one. There's at least one, but we don't 100% know what it is. So it is very likely multiple things. So these are broad scale trends that are controlling leaf shape. So specifically toothiness. So there is the freshwater margin effect. You tend to have more leaves with teeth along the margin of rivers, etc., because their feet are wet and they need to let the water out.
Yeah, they need to let more of those points for the
for guttation.
Yes.
Yeah,
exactly.
Because you're hanging out where the water is.
Exactly. So in general, you tend to have a higher proportion of toothed leaves than you would expect for a given temperature in places where you have basically higher water tables. Another idea for the leaf teeth is that it could help with early season photosynthesis. So basically, you can increase the length of the growing season. Basically, you can start photosynthesizing earlier in the growing season with increased sap flow. So again, you're moving things through the tubes. So these are areas of increased photosynthesis early on. That one is probably not the only one. A similar like root pressure. So if you have teeth, you're continuously moving the sap through the tubes. And so this prevents an embolism. This prevents a bubble, right? If you have this continuous flow. So again, that's probably important. I mean, it's definitely important because like if a plant gets a bubble in the tubes, it cannot use the tube.
Right. Yeah. That's bad.
Yeah, that's bad. You cannot use the tube anymore. But it's probably not the only thing. My favorite reason, and I think it's probably in combination with other things, but what I think the most convincing reason is bud packing, also known as vernation. So for plants that live in places where they have to drop their leaves, so the northern hemisphere, this is temperate regions because it gets cold. The plant makes the leaves and hides it in a bud over winter. So it makes the leaves in the fall, and they hide out in their bud over winter, and then in the spring they flush. So the leaves are easier to pack into a bud if they are toothed.
Yeah.
They're one of my favorite papers was looking at, so origami is folding paper, kirigami is cutting paper. So they basically were looking at leaves with respect to kirigami and figuring out like what is the best way to fold the most leaf into a bud. It's because it's fascinating. If you look at baby leaves, little bitty baby leaves, they are super toothy. They have more tooth, like they're mostly tooth. Which makes sense because like you can fold those in on themselves very well, wad them up tight, because if your bud is too big, it might freeze.
Right.
So if you have a little bitty bud, it'll survive the winter.
So those that once again, it's physics.
Yes.
In this case, it's geometry.
Yes.
Now it's pure maths, where the shape is just a better shape for packing into that condensed form that you need to start out in.
Exactly. And the other fun thing too is like getting into the like phenotypic plasticity. So plants will have leaves, so preformed and neoformed leaves. So preformed leaves, they make them the year before. Neoformed leaves, they make the year of.
Sure.
So they actually are getting two different temperature and precipitation signals.
Oh.
So
You
they
get
have
two different types of leaves?
You do. There's slight differences, but yes,
Cool.
basically you need to know about what node was where they formed directly and which ones were in buds. But basically, yeah, if they were in buds, they are reflecting the previous growing season because that's when they were made. And the ones that form directly reflect the current growing season because that's when they were made.
That's cool.
Neat.
So this is a trend, it sounds like we see where the shape of leaves, which is so important to the things that leaves are doing and which we think of as being so iconic to whatever plant it is, will vary quite a bit just from year to year, just as over the lifespan of that plant responding to the environment.
Yes.
But it sounds like those changes and trends in leaf shape are consistent across many different groups of plants.
Yes, they do tend to be. The specifics tend to be like group specific, but in general, but also within a family, not every genus will have phenotypic plasticity. Within a genus, not every species will have phenotypic plasticity. And there are other ways to have phenotypic plasticity because, so we're talking about between growing seasons, but oaks are famous for the variability in leaf shape in a single tree. Because you have sun leaves and shade leaves, and they are different sizes and shapes.
Yeah.
Because the sun leaves tend to be smaller and, lobier, toothier because they don't want to overheat. And then shade leaves tend to be much wider and smoother because they're trying to maximize all of the light that is actually percolating through the canopy. So like that, like even within a single tree, you can get profound variation in leaf shape.
Yeah. And that's not, we were talking before about how you have plants where there are leaves have been adapted into parts of the flower or leaves are over here for different functions. But this is variety in leaf shape for leaves that are doing the same job.
Yes.
Under slightly different conditions.
Correct.
Which is very cool.
I love them. This is what I get really into. Like this is the part where I'm really trying to stay on the rails.
Well, while we're on the note of things that you're really into, let's talk about fossils.
Yes.
Starting with the question of obviously we get fossilized leaves.
Yes.
What do leaf fossils how do leaves tend to enter the fossil record?
I'm going to give you an unhelpful answer and then I'll give you a better answer. They look like leaves.
They do look a lot. Leaves are some of the best fossils.
Absolutely.
For like education, especially.
Yes.
One of the things you run into, and this happens a lot with vertebrate animals, is you show a thing, you're like, this is a bone, and people go, is it though? Because that looks like it's a rock.
Yep.
And you're making things up.
Yes.
But when you go, a leaf fossil, that's a leaf. That is a leaf that is part of a rock somehow. That is a really visual, intuitive demonstration of what a fossil is.
Yes.
Absolutely.
And even when you have like a bone that is recognizable as a bone, it's still having to be like, I promise you this is the shin.
Yeah, this is what a tibia looks like.
Yeah.
Yes.
This is what the shin of this weird animal we don't have an example of today. So I can't actually show you one. We drew one. That's as good as you can get. Yes. A leaf's a leaf. And everyone has had an experience with a leaf at some point.
Yes.
So it's like, I've seen that in my backyard.
Yeah.
Boom.
It has the same sort of immediate recognizable appeal as like an insect in amber.
Yes.
Absolutely.
Like, I've seen an insect before. This is a rock with an insect in it.
Yes.
Leaf fossils are very, very iconic type of fossil.
Absolutely. Like, I will argue forever that they are some of the best fossils for outreach for that exact reason. The amount of imagination it requires to be like, this toe like is from a mammoth.
Yeah. Because so often you're seeing a bone you've never even heard of before.
Exactly.
You didn't know there were that many bones in the foot.
Right.
Right. Exactly. Versus this is a leaf. One of the outreach things that I would do during my PhD was we would take fossil leaves from the San Juan Basin of New Mexico. So they were Paleocene. They're like 65 million year old leaves. And then we would have modern relatives. Yeah. And we'd ask people to match them up. And it's so easy. Yeah, like, yes, this is the modern, this is the fossil. And it was really, really cool because when I would do field work in Kenya, like it was so easy to get people engaged because like, look, it's a leaf. That transcends any language barrier.
Yep.
So in general, leaf fossils look like leaves. And one of the things that I really want to get across, one of the things that I really want to make sure that people understand is that leaf fossils are so common in the fossil record. It's my favorite thing is to talk to people about sample sizes in paleontology, because like, vertebrates, you get one bone, right? Versus like, I was dealing with hundreds of things. I have colleagues who have thousands of things. And oh man, and it makes sense, right? Think about how many leaves are on a tree.
Yeah, it's not just one tree.
And you can do it multiple times.
It's the shark tooth concept.
Yes,
And why are shark teeth so common? Well, because one shark has hundreds of teeth and constantly makes new ones over the course of its life. A single shark can generate several hundred teeth.
Right.
To the fossil record. A tree is the same way. A single tree is going to have hundreds and hundreds of leaves.
And especially if they're dropping them.
And
every season.
They keep dropping them.
And you tend to not just have a solitary tree. You tend to have a forest.
They move in herds.
So, in general, the most common fossil types for leaves are compression and carbonization, which makes sense because they're flat. Like, that's the thing. Leaves are already flat. They are primed for fossilization. So compression is basically the taphonomic equivalent of you put a leaf in a book, right?
You're pressing a leaf between sediment.
Exactly, forever. Those can be... Often they can be just impressions, right? So you just have the shape of the leaf. Sometimes you'll have carbonization, so you'll also have organic material, carbon left behind. You'll often have preserved cuticle. So leaf wax. I am very bitter because my leaves from Kenya did not have cuticle, but all of my friends' leaves from North America did, and I'm just very bitter because I didn't get to have cuticle. But in general, that also means that like the type of prospecting, the type of excavating that you do for fossil leaves is fundamentally different than what you do for say vertebrates.
It sure is.
We move hillsides. The amount I calculated at one point, the like square meterage of hillside that we removed for my PhD, and it wasn't like, it was barely anything compared to some of my colleagues, and it was considerable because, as my colleague Peter Wilf once said, you can't get big leaves out of small rocks.
Yes. Well, and I think that it's valuable to make that point that the conditions that preserve leaves, and this is true for a lot of plant stuff, are not always the conditions that best preserve animal remains. And the way that you go collecting those things is not the same
correct
method. So if you want to find good fossil leaves at your fossil site, you need to actually, we've all worked at the Gray Fossil Site.
Yes.
And there are leaf impression fossils known from the Gray Fossil Site. There's a bunch of really nice ones. The way we dig is not recovering those leaf fossils because we're looking for tapers and turtles and stuff.
Right. So when you have a really nice fossil site where you have like a bone bed, right, you're going meter square by meter square, you're going down uniformly and you're scraping away, that's going to destroy every leaf.
Yep.
Because what you want to do if you were trying to get out fossil leaves, you're basically taking out big chunks. So you have these laminated layers, right? You turn it on its side, you hit it with a rock hammer, and it will break at the point, the weakest point, which tends to be where the leaf is, and it'll crack open with a leaf inside. And you lather, rinse, repeat hundreds of times.
Yes. Until you have a whole tree.
Yes, exactly. So in general, leaves tend to be very common fossils. They tend to be very charismatic fossils, and they can be incredibly informative fossils. So in general, the earliest leaves tend to look less leafy. Like I was kind of talking about, those earliest leaves, especially the earliest microphyll leaves, are basically extended shoot nubbins. So those are way less charismatic. It's basically like, oh, lumpy twigs. But they're important lumps. But yeah, in general, leaves are really, I love showing off leaves. Like Maison Creek fossils in particular, like the ferns from Maison Creek. You show that to someone, you're like, yeah, that's a plant. I love it. That's the reaction I want.
Allie, this next question I ask knowing what this question is going to do. But since this discussion has been so short, I figured we've got opportunity.
Let's go.
What sorts of things can we learn by studying fossil leaves?
I have given hour-long talks on this topic.
Give them all right now, back to back.
Okay. So this is what I do.
This is your research. This is so for all of the Allie fans out there who have been listening with the podcast for years and years and years. Allie has been on the podcast over a dozen times. We have alluded several times to your research.
Yes.
What can we learn from fossil leaves is your specialty.
Yes. This is the thing that I want to talk about all the time.
Go ahead.
Oh my goodness. I'm so excited. All right. All right. So in general, I'm going to kind of divvy this up by the type of question that can be answered, and then kind of show how we can answer it. So in general, the 4 categories that I will be mentioning are paleoclimate, paleoenvironment, paleoecology, and paleoatmosphere.
Nice.
Full disclosure, I don't mess around with the atmosphere, but I will talk about it. So paleoclimate in particular is one of my babies. Oh, I love it so much. I have mentioned it already this episode. So this is the relationship between the margin/shape of the leaf with temperature and the relationship between size with precipitation. So this is only for woody dicots. And this is amazing. This is absolutely wild that if you can have at least, say, 15 to 20 different species, and I'm using heavy air quotes with species because we don't actually assign fossil leaves to species because, as I mentioned, leaves are the worst way to identify a plant.
Yeah.
We use what are called morphotypes, which are basically, these are different shaped things. We think everything that is this shape is probably the same thing and is probably different from the other things.
It's kind of a footprint situation where
yes,
we can definitely get large groups and have like, it's probably one of this handful of things.
Yes.
But I can't say who.
Yes, exactly. So this is something that I think about. This is my Roman Empire. This is the thing that I think about on a daily basis. It's the relationship between leaves and their environment. And so when we talk about these patterns, right, we tend to have a higher proportion of toothed leaves in the northern hemisphere, we are specifically talking about the temperate northern hemisphere. So we have known these patterns for more than 100 years. So the first paper that was published on this, there were 2 of them back to back that came out in 1915 and 1916. And that is the basis for this sort of paleobotanical research. But these were some Europeans who were looking at trends in Europe, and they hold true in North America and in Asia, but they do not hold true to the same extent in the tropics or the southern hemisphere. The number of things that we have models that are based on northern temperate trends that are not really applicable in the tropics or southern hemisphere is annoyingly high. So in general, and part of the reason for this is like the distribution of plant groups. So you tend to have trends in like leaf shape in different plant groups. And some groups are found all over the world, and some of them are only found in the southern hemisphere, some of them are only in the northern hemisphere. But our models tend to not include the groups in the southern hemisphere. Which means that we are missing out on these swaths of the diversity of shape. In addition, keep in mind the difference in the shape of land masses on the planet. So the northern hemisphere has wide continents and the southern hemisphere has tall continents, which means that you don't have the same kinds of continental interiors in the southern hemisphere.
Yep.
Which is going to have impacts on, the ways that, plants deal with specifically precipitation. One of the things that I am actively looking into is trying to incorporate more of the Southern Hemisphere, specifically tropics, specifically Africa, into these models. Because, like, for example, the model that we used for my dissertation research did not include African floras. And I was working in tropical Africa, so I had to make my own.
Yeah.
Because they didn't exist. And it's incredibly frustrating. Another weird, frustrating thing is that I kind of mentioned this before. So when in the fossil record, unless you can find a compound leaf all associated, you do not know that you are looking at a compound leaf. Sometimes you're really sure, but you haven't found it yet, so you can't really do anything about it. So that means that when we are doing our estimates for leaf size and precipitation, we treat simple leaves as equivalent to leaflets. So leaflets is equivalent to simple leaves. And I am so sure that that is incorrect. Because, like, when we're talking about the functional unit of the leaf, the area that we're talking about, I'm so confident, has to include all of the leaflets, which means that we are underestimating the actual area of these leaves, and who knows how that's messing things up. Would you care to guess what part of the world has the highest frequency of taxa with compound leaves?
Oh, is it your part of the world where your research takes place?
Yeah, the tropics and the Southern Hemisphere. Yes, that would be it. Which again means that it has not been previously considered. And so we don't really know how our estimates are being biased by the fact that we don't know whether or not we're dealing with a compound leaf or not. So that was my soapbox. I will go a little bit faster through the other things, but that is what I study. That is what I find most interesting. I would talk literally all day about this.
Sure. Well, so the sort of premise of using leaves to reconstruct ancient environments is very straightforward. That we talked about, you've got all these different shapes that leaves take based on what sort of environment they're living in, based on what sort of environment they're growing in. So we can look at fossil leaves, see those similar structures, make inferences about those environments.
Exactly.
But I think that your point there drives home the importance of understanding all of the factors that go into that. And this is one of those really interesting emerging sort of manifestations of that geographical bias historically in paleontology. We've talked a lot on the podcast before about how, we know a bunch more about the fossil record of Europe and North America than we do about, say, South America or Australia, because Europe and North America is where all of the scientists historically have been living and doing their work. This is a really interesting case where even when we do have the fossils from places like Africa or South America in the tropics, science also has historically not done as much work understanding the modern plants in those sorts of regions.
Yes.
So there's less we can do with those fossils without that information.
Correct. Like that, what is it? Like diversity blind spots. So places with high diversity that aren't necessarily well represented in the literature. Africa is one of them, and South America is too. Though paleobotanically, we're doing great with South America.
Oh, good. Well,
but
fantastic.
The funny thing too is that so my research, I collaborated a lot with paleoanthropologists. So like I was helping work on the context for human evolution. And the fact that, this is something that people are so interested in, but we do not have what we need to be able to accurately talk about this is incredibly frustrating. And part of it is because the preservation that you need for leaves is different than animals. And the people who are looking for the teeth of hominids are not necessarily going to be looking out for, fossil leaves.
The teeth of leaves.
The teeth of leaves, exactly.
Yep.
So they might acknowledge, like, oh, I saw some plant bits over there. But the type of really methodical collection that you have to do, it takes multiple days to get enough leaves in order to do these sorts of analyses. And, it takes a lot of staring at leaves to be able to like differentiate them. So that's not something that has been done historically. And I think that's a an absolute shame. Let me do it. So to finish up on paleoclimate, you can also use carbon isotopes. So again, sometimes you have cuticles, you have carbonization, so you can use carbon isotopes. That's related to water availability. So you can use carbon isotopes to get a sense of, what was the hydrology, and that you can use on any vascular plant. So that's just not just woody dicots. If you got a plant, you can look at the isotopes, because fractionation. So again, it's just physics, right?
Mm-hmm.
It's water moving through the plant, and some water is heavier than other water.
Yep.
So another thing that I'm obsessed with is paleo environment. This is what I was kind of talking about earlier with the leaf lifespan, the leaf economic spectrum. So basically, this is a whole paradigm, this whole thought process that looks at like leaf thickness and seasonality and getting to carbon to nitrogen ratios. So like there's a whole lot that you can do that with that. In general, I'm going to simplify this. So in general, the petiole width, so the width of the leaf stalk, is related to leaf lifespan, because in general, the longer the life of a leaf, the thicker it's going to be, right? If you're going to have a leaf for a really long time, think about magnolia leaves. Those are leathery.
Mm-hmm.
So if you're going to have a leaf for a long time, you will put resources into it. In order to hold up a heavy leaf, you need a thicker petiole.
Yep.
It's just physics. And because we cannot measure the leaf mass per area of fossil leaves, because they are compressions, you might just have the impression of the leaf, right? You can't measure the mass of a leaf that isn't there.
Yep.
Just in general. So instead, we look at the width of the petiole. That'll give us that is related to the thickness of the leaf. That is related to leaf lifespan and deciduosity. Right? So in general, thicker leaves tend to live longer. They tend to not be deciduous. You tend to hold on to them. So thinner leaves tend to be deciduous. They tend not to persist as long, because if you're only going to have this for a growing season, like why put a lot of resources into it?
Yeah, that makes sense.
And so you can use that to get at seasonality. And there are different ways of being seasonal. Like, we in the temperate northern hemisphere, we think about, oh yes, winter. That is, when all the plants are dormant. But in the tropics, you will have tropical deciduous forests, tropical seasonal forests. Basically, they are dropping their leaves because it gets too dry.
Yep.
So if you have leaves, you can look at the width of the petiole and figure out the paleo environment. You can figure out, how dry is it. And so that's just woody angiosperms. One of my favorite things is if you have cuticle, you can look at the shape of the epidermal cells. And that will tell you how much light the leaf was exposed to.
Ooh.
Right? So they're all kind of squiggly looking. But in general, they use the undulation index, which means that the wavier the cell margins, the lower the light that the leaf was exposed to.
Gotcha. So you could potentially tell, like, is this high in the canopy or is this deeper in the forest?
Exactly. So this is a proxy for canopy cover. So how open or closed an environment is.
That's very cool.
Which is wild. So this is primarily, if you do it from cuticle, you can do this. This is only for woody dicot angiosperms, but sometimes you can use phytoliths, which are plant rocks. So silica bodies that are in many parts of the plant, including leaves. Sometimes you can use the shape of phytoliths to get at this undulation index, and that will allow you to include more different types of angiosperms, which is really cool.
That's cool.
Is the undulation, like, is the squiggliness, like, increasing surface area for light absorption, or is it?
I imagine so, because they tend to be wavier in low light.
Yeah, so that's what.
Yeah, so that would make sense. I'm not 100% sure, but
the waviness confuses the light waves and they get trapped.
Yes.
Well, it's trying to emulate it.
Right.
Like, I too am a wave,
and also
we should hang out.
Yes. And then lastly, this isn't necessary. This is something you can do with leaves, but it's not something that you can do with leaves. I'll explain. Is lateral heterogeneity. So basically, I collected a whole bunch of leaves, and because leaf fossils don't tend to be, they don't tend to travel very far. They tend to be very close to their source because leaves are fragile.
Mm-hmm.
Right.
And so they tend to be very close to their source. So you actually get a very good sense of the pretty much standing diversity. So it's not 1-to-1, but you can see patterns. And so one of the cool things that I saw at one of the fossil sites that I worked at was that there were these in situ stumps with roots coming out. So we knew there was a forest over there because we have it.
Nice. You have the bottom of it.
We do. Yeah, exactly. And so there were leaves near that. So the leaves near that, near the stumps, tended to be the leaves of woody dicot angiosperms, which makes sense. Those are the trees they came from. When you got farther from where we found the stumps, you still got leaves of woody dicots, but you also got monocots and emergent aquatics. So suggesting that there were like openings and maybe ephemeral ponds. So like we can get a sense of the actual landscape from leaves.
That's because this is like a thing that you have reminded us of many times in many episodes. Plants don't move.
Yes.
Those leaves are where this is an issue we run into with animals.
Yes.
Where it's like, we found this animal here. Is this a typical place for this animal to be?
Yeah.
Did it get washed down a river or something weird?
Did you live here? Were you passing
through? When we find an ankylosaur in the ocean, it's like, this is not, you did not fall where you died.
Right.
This is an unusual place.
Yes.
With the idea that you could have a broad fossil deposit and say that's where the forest was and here's where the meadow was.
Uh-huh.
Is very cool.
Yes, it is. It feels like time travel.
Yeah.
In a way that you don't often I like to joke that paleontologists were time travelers, but like that specifically is way closer to time travel than any other thing that I get to do.
Well, you don't usually get to make a local map
exactly
of the environment you're digging up.
Yes, exactly. No, it is. It's kind of wild.
Yeah.
To get and you can do this with leaves. You can actually do, if you do have some fossils, you can do that sort of thing. But yeah, just getting this physical sense is really fun, and that kind of goes into the paleoecology. So that's one of the more straightforward things that you could do with fossil leaves. It's just counting, that's what it is, and that is any plant. Like if you have a plant, then you can count it and you can figure out how many different types of things there are. So this is diversity indices and species richness. And this can be really helpful if you're trying to compare through time or across space, different types of environments. But what I like to do, which is also just counting, but it's harder counting.
Advanced counting.
Having flashbacks to those lemna leaves again.
I honestly... Kinda. So, so you have these leaf morphotypes. And so you have to have the morphotypes first, right? So these are the different units that we're counting. How many different units do we have? If you are lucky, and you're not always when you're dealing with fossil leaves, if you are able to look at things, look at morphology and get a sense of the higher order taxonomy, right? So if you have a grass, you can actually be pretty confident that you're in the family. Even if you can't necessarily say what genus or species it is, you can at least narrow it down to family for a lot of groups. And sometimes you can get more specific. Like there are, if you have this higher order venation, you might be able to look at that and get a sense. But even just at the family level, one of the things that you can look at is community composition. Mm-hmm. And you can get a sense of, because a forest is not a forest is not a forest. There are different types of forests.
Yes.
There are different types of plants that have different shapes, that different roles in environment. And it is fascinating that even within, say, tropical Africa, the community composition, the families that are actually making up the flora can vary dramatically. And that's going to have a big impact on what the leaves look like, because you have these family relationships that are going to impact leaf shape, but also that's going to impact who can live there and who can eat things and that sort of thing.
Yeah.
And you can see these really cool trends. So for example, with the Gray Fossil Site, our favorite place, if you look at the community composition, if you look at the groups that are actually there, wow, it's not really that different from what you see in East Tennessee today.
Right.
Yep.
I like to describe it to people when I'm talking about it that if you flew over the gray fossil site back in the day, you would not have noticed.
Right.
So, yep, those are oak and hickory and pine trees, and there's some birds and stuff, and then you could just continue on without taking a closer look.
Exactly. I like to joke that if you were in a time machine and you got out, in the Neogene, you would not necessarily know that anything was different until a tapir walked by.
Yes.
Then you would know.
Well, and I know another cool aspect of paleoecological interpretations with leaves specifically is that leaves can also bear trace fossils.
Yes.
Not to bring the animal side of this into it.
I see how it is.
But I know, and actually you mentioned Peter Wolf before. This I learned from Peter the first time I had ever learned about insect feeding trace studies on plants, that a leaf fossil can tell you not only what kind of plant was there, but what kind of insects were there, because the leaf is from the plant and the little bite marks are from the insects.
And it's wild to see. So you can do so much with the diversity in like tracking the types of feeding traces, right? Specialists versus generalists. Like there are some things that there are some types of feeding that don't show up until certain points. It's an innovation. Unsurprisingly, you tend to see more generalists after an extinction event.
And that was actually the research that Peter was doing when I heard about it, was leaf feeding traces on leaves in and around the end Cretaceous extinction, and seeing what changes. We lost certain types of leaf eaters.
Yes.
In that extinction event.
Exactly. One of my colleagues, she has one of the best names. Her name is Gussie McCracken.
Fantastic.
I know. She is a curator of paleobotany at the Denver Museum. So she studies, these plant-insect interactions. So she's the person who gave a talk about fossil Acarodomatia. So these fossil mite houses on leaves. And I remember being at that talk at GSA, and the paleobotanists were going wild. Like people were asking all sorts of questions like, oh, how do I recognize this? I want to go home and look at all my leaves. It was very exciting. And that's, again, like another thing that you can get at is like looking at the diversity in these Domatia. I just looking like, how long has this relationship persisted?
Yeah.
Also, to just less directly connected to animals, feeding and leaving traces, you're saying the types of forests are different, and looking at what kind of assemblage you have. Animals react to the environment they're in, and the environment they're in is made up of the plants that are around.
Correct.
So, like, that's important to know. Was this predator in a forested environment? Because that is going to tell us what kind of ambusher it might have been. Or was it in an open environment? Does it show leaves from an open space? That would be a pursuit.
Well, and I think that it's really interesting, and this is not within the scope of this episode to go into, but the amount of animal adaptations that are reacting to leaves.
Exactly.
That the obvious one being teeth.
Yes.
Right? Animals that eat leaves have specialized teeth or mouth parts if they're invertebrates. But also, we've talked about on the podcast before, animals that live in forested habitats tend to have different color patterns on their body, because the light is mottled, and it's mottled because of leaves.
Yes.
That's what's up there modeling the sunlight.
Yes.
Is leaves.
And you get all the way up to mimicking and camouflage.
Yeah.
That actually is going to help you blend into this leafy
Yeah.
So often when we talk about a new species of animal, we're like, and we think it lived in this environment. That's what all of this is understanding what kind of environment by looking at the plants. So it is critical to putting animals in context in the fossil record.
Exactly. One of the things that I have to say a lot is plants are not just your green background.
Yep.
So one of my greatest pet peeves is, I'm so sorry, when animal people try to use animals to say what the environment is.
She's talking about us.
Those jerks.
Those jerks.
There are multiple paleontological studies that have attempted to use the size of fossil snake vertebrae to interpret paleoclimate conditions.
Yep.
This, I assume, is exactly the sort of thing that Allie's complaining about.
Okay. This is okay. I applied for a postdoc I was really sassy in my letter of interest, to the point where my advisor are you sure you want to say this? And I said yes. And I got a follow-up interview because he was intrigued by how sassy I was. Where basically
you heard it here, kids.
Yes. So basically, there's a lot of research that looks at gear ratio in the ankle of ungulates to get a sense of if they were living in an open or closed environment. Basically, are you made for speed or are you made for maneuvering? Do you need to be able to like get through a closed canopy versus like, oh goodness, I'm on the savannah, I just gotta run. And the distinction of open versus closed environment infuriates me. Like, it is because it's a gradient, right? Between like, oh goodness, I can't walk through here, to this is open completely. There's not anything, on the horizon for miles. And so I get very frustrated by the neatness of the dichotomy of open environment, closed environment. But unfortunately, it's really hard to do anything about that because plant and animal fossils tend not to be preserved together.
Yep.
Yeah. And this is why those studies come
and that's why this happens.
Is because you say, well, I'd love to know what the environment of this place is, but either because of the preservational conditions of this deposit or because of the way we excavated,
yes,
I don't have plants.
Yep.
All I have are these animal bones, and we gotta see what we
gotta
can
do
tell.
something with them. There was a paper that came out in I think the 80s that was a botanist and a mammologist, and the paper it has since been added on. It's like, okay, it's not as neat as this laid it out, but it was basically looking at modern habitats and looking at the shape of the forest, right? So where there are lianas, how far apart were were the trees, that sort of thing, of tropical rainforests, and looking at the locomotion of the mammals that live there. So, like, do you have a lot of gliders? Do you have a lot of things with prehensile tails? Like that sort of thing. How are they moving? And I think is what we need more of. Are these modern collaborations between, botanists and zoologists to get a better sense of the relationship between modern plants and modern animals so that we can better apply them instead of like, again, thinking of plants as the green background and not really parsing like what it means. I feel very strongly about this.
I really appreciate you making the point that it's a gradient.
Yes.
Because that's something I'll think of often when I see descriptions of closed or open habitat. It's like, I've been in forests I can run full tilt through.
Yes.
Like pine forests where the trees are just telephone poles because all the branches are up top.
Yes.
And I can't reach two trees with my arms.
Right.
Like, I've got 8 feet between them. You can just run through there easy.
Right.
So that wouldn't necessarily show adaptations for a forested habitat, but you're absolutely in a forest.
Exactly. Okay. One last thing before we move on, because, oh my goodness. So, atmosphere. PCO2. This is not something that I personally study, but it is incredibly helpful for, again, understanding the context that, plants and animals are living in. So, plants have this really close relationship with CO2. They're basically besties. And so there are a lot of different ways that you can try to use plants to estimate paleo CO2 levels, so PCO2. So you can do it isotopically. That is debatable how accurate that is. Another way that a lot of my colleagues use is the gas exchange model. So basically, it's physics. So looking at the isotopes and also looking at the stomatal density and the size of the stomata, it's really important that you have that calibrated to your specific plant group, though.
Yeah.
So you can do it on any plant group, but you have to have a calibrated model. So like a colleague of mine, he was working on sycamores. So he grew modern sycamores and then applied that model to fossil sycamores, but you wouldn't be able to apply the sycamore model to an oak.
Yeah. Cool. I like that, comparing, like, here's what the chemistry says, but also here's the shape of the passages that these molecules are moving through.
Yes.
with those two combined, we should be able to get the math of the rate or the amount, the density it's happening in.
Yeah. With our powers combined.
Well, and that method, that proxy for understanding carbon dioxide levels through time, it's funny to go through and talk about these things because so many times on the podcast we have mentioned, here's this time period, here's this event, here's this habitat. Oxygen levels, carbon dioxide levels, temperature, precipitation, all these things that have been estimated that we so often mention very briefly as, yes, here is what this habitat is thought to have been like.
Yeah, just background stats.
Yes.
This is where a lot of that information comes from. Sometimes it's coming more directly from like seeds or pollen or whatnot, what types of plants there are. But leaves are this fantastic measurement, these devices that record within them aspects of, they're these little weather vanes, these little, they're just recording the climatic and surrounding conditions.
They're barometers.
Yes. Very neat.
Allie, I'm going to ask you one last broad question about leaves, and it is the question that I have asked you many times on many of your plant episodes because I think it's a fun question to ask, and I think that it really drives home the point that we like to talk about the sort of foundational importance of plants in Earth ecosystems. Allie, how did leaves change the world?
Okay. So when I was asking all of my colleagues to give me their favorite leaf fun facts, one of the curators said, "It's the only organ you can see from space."
Not wrong.
Which is so important. So often when I give talks, I will start out with a picture of Earth and point out, yes, we are a blue planet, but we are a green planet.
Mm-hmm.
And when we are looking at that green of the planet, you are looking at leaves.
Yeah.
And just think about that. Like, I feel like that could be enough. Like, mic drop, that's it. That's all I need to say.
Yeah.
Well, it's if aliens were to look at Earth, one of the only organisms they'd be able to actually get decent information about are the leafy plants.
Yes.
Earth is water and leaves and clouds.
Yeah.
Yep.
And ice, I suppose.
Yes.
There's some ice on it on a clear, if it's nice and clear.
Mm-hmm.
Yes.
That is a phenomenal point. That is incredible. Well, and then it's a great point because we think of, plants are green, but specifically, leaves are green.
Yes.
If I just get you a random part of an oak and it's not a leaf, it's not green.
Yep.
It's bark colored and it's wood colored.
Yes, exactly. So the innovation of leaves changed the face of the planet.
Yeah.
Changed the color of the earth.
Yes.
Which, man, what must have Earth looked like before?
Brown.
Like, just brown and...
Brown and blue.
That's
so weird.
I know, right?
Well, and we talked, I think, about in the photosynthesis episode 145, that photosynthesis is the process that allows ecosystems to exist as we know them. And we talked, we did a whole episode about herbivory, episode 173, where we talked about just the fundamental shape of the food webs of Earth. And leaves are the things that are doing the photosynthesis.
Yes.
So all the importance of photosynthesis for terrestrial environments comes down to the function of leaves.
Yeah.
Yes.
Without photosynthesis, our planet could not utilize the nearly infinite energy being put out by the sun.
Yes.
And leaves are the structure doing a extreme portion of that photosynthesis.
So without leaves, you don't have vascular plants, right?
Yeah.
Like, I mean, it kind of goes the other way, but in general, and then that would have a major impact on the diversity of life on land, just in general, right? Because it is the innovation of leaves that allowed trees to happen, right? Like the whole face of the world changed partially because of leaves. Like the origin of vascular plants, that predates leaves, but in general, they had this huge diversification event after the origin of leaves, precipitated a 10-fold increase in plant species numbers and led to an 8 to 20-fold increase in atmospheric CO2 drawdown, significantly shaping Earth's geosphere and biosphere.
Yep. Yeah, when we talk, we talk about stuff now with like technology. It's funny how we keep comparing technologies. That we talk about solar panels, a technology that can harness energy from the sun, and we talk these days. It's very common to talk about methods of carbon dioxide drawdown. Can we get the carbon dioxide out of the atmosphere? And leaves are doing that all over the world. Yes. That is the technology of leaves. That is what they do.
Yes.
Is they harness energy from the sun and they pull CO2 out of the air and they make oxygen.
Yes.
So they are, we've talked before about how plants have terraformed the earth. That's what plants do.
Yes.
And of course, the roots are an important part of that, in many, but leaves are the main way that plants change their environment around them. They are just a bajillion little units of altering their surrounding environment.
Exactly. And you mentioned so that leaves are, plants are giving, you're taking down the CO2, they're giving out the oxygen, but you can't forget what they're doing with water. Right? Because they're just tubes. And you talk about how the rainforests, the lungs of the planet, that's because of the leaves, right? It's that is what is releasing the water into the atmosphere.
Yep.
This is the part that's blowing my mind.
Yeah.
Well, because it's so, and you sort of mentioned it before, all plants are doing the same thing.
Yeah.
And they're all doing it with leaves, which means that this is one type of organ that is responsible for all of this stuff.
Yes.
That's extraordinarily cool.
Yeah.
Allie, I hope you have had fun on your Leafs episode.
Oh my God. This is the best thing that's ever happened. I'm so happy.
You heard it here, listeners. It's all downhill after this. We'll see. We'll do a Leaves part 2 at some point.
Oh, no. I would love that.
There will be no outline. We'll just invite Allie and be like, all right, Allie, we're setting a timer. Go. Say stuff
dangerous.
About Leaves.
Oh, dangerous, but wonderful.
We'll do it'll be like the end of the year Q&A, but it's just all Leaf questions.
Yes.
For Allie.
Yes.
Before we wrap up our whole episode, one last thing to do, and that is our patron questions. Every episode, we read out some questions that were submitted by our patrons. This is something that patrons can get access to by subscribing at a certain level on the podcast. This episode, I've got 2 questions here. The first one was addressed in this episode. Marguerite asks, are there chloroplasts in all plant cells, including roots and vascular tissues? And if so, why aren't they green like leaves?
So I did mention this earlier, but I will summarize it again. So basically, if it's not green, it does not have chloroplasts. So in general, that tends to just be in the leaves. But any other organ that they use for photosynthesis, so like cacti, they're photosynthesizing with their stems. Again, this goes back to when we're taught about, this is what a plant cell looks like. Apparently only ever showed leaves, which makes sense. I totally understand. And then bear in mind that not all leaves have chloroplasts because not all plants photosynthesize. Mm-hmm. So it's kind of both sides. So not all cells, not all plant cells have chloroplasts. That's why they're not green. But not even all plants have chloroplasts, which is also why they're not green.
Yes. Yeah. A topic for a whole other episode.
Yes.
Things like parasitic plants.
They're just wild.
Wild stuff. Are there any plants that photosynthesize with root tissue?
I am so confident the answer is yes.
Yeah.
Okay. Me too.
I don't know. I don't know off the top of my head.
Okay.
But I'm so confident the answer is yes. I cannot confirm.
It kind of must be, right?
Surely there's an aerial root somewhere.
Yes, exactly. That's kind of what I was thinking about, like bromeliads.
Yeah.
They're, if you have your roots out there, just maximize photosynthesis.
If they're gonna be in the sun, then yeah.
Here is our second patron question. This one comes from Emily, who asks, are there any botanical citizen science efforts that you would recommend to plant enthusiasts?
Ooh.
Absolutely. This was fun. I had to stop myself because I went too hard. So
we can also put a bunch in the blog post.
Yeah.
So you can mention your favorites.
Okay, cool. So First and foremost, there are a lot of places that you can find huge lists of citizen science projects. So if you are in the United States, the US government has a website for that. If you are in the EU, they have a website listing a whole bunch of them. So here are some that I pulled out because I really like. So there is Project Budburst. So there is an app associated with this. It's through the Chicago Botanic Garden. So basically, the quest is to document plant and animal interactions, monitor plant life cycles. So that's where Project Budburst comes from. So when are they fruiting? When are they flowering? When are the leaves flushing? Basically, just to get a sense of, hey, what's going on?
Mm-hmm.
Similarly, there's Nature's Notebook, which is part of the USA National Phenology Network. So phenology is the timing of things. So again, you can look into things like that because, with climate change, the timing of fruiting and flowering is changing because some things are related to temperature, some things are related to light. So being able to monitor that. And the nice thing about plants is they don't move, so you can actually monitor the same it's really easy to monitor the same plant over multiple years. You don't have to stick a tracker on them. You just gotta remember where you left the plant.
Yep.
Similarly, doing iNaturalist observations is very helpful, so we can get a sense of who is where. Fun Leaf is, I was gonna say fun, but yes.
Sure.
So it's basically, it's looking at the biodiversity of organisms associated with plant leaves.
Okay.
So this can be fungi, bacteria. So, and this is based out of Europe. So basically, doing sort of DNA analysis. So taking samples, sending it there, so they can do environmental DNA to figure out who's on this leaf. And then one of my favorites is Notes from Nature. So Notes from Nature is, so most of those were all going outside,
Yes.
right? Going and meeting the plants where they live. Notes from Nature is more inside. So it is transcription, often of herbarium specimens. So herbaria have way more things than we can database on our own.
Sure.
So what Notes from Nature allows you to do is look at photographs of herbarium specimens, so these pressed plants, and you basically transcribe the label data, because that is something that if you can do that, then we can add the data to our databases, add it to data aggregators, and make things more available for people all over. So if that is something that is of interest to you, I highly recommend it, especially if you know languages that aren't English and if you are good at reading cursive.
Yeah.
So a lot of these are typed out, but some of the hardest transcription, especially for machines, are things that are written in cursive.
That
because
flowery Victorian
oh
script.
my goodness.
Yeah.
I have looked at so much handwriting. I'm really good at knowing what country someone was from based on their handwriting, which is a really niche skill. It only works from the last century. But.
It's like those fancy Victorian scientists weren't even considering AI reading their handwriting in the future.
Sometimes I don't think they were thinking about humans reading their handwriting in the future, because a lot of what I do supervising students is coming over and they ask me, what does this say? Because everything, especially when everything was handwritten, people came up with all sorts of unique shortcuts.
Yeah.
So that's why you have William, which is WM. We don't need all those words in the middle. And also, if you know any Latin, that's really helpful, because especially old-timey botanists, they love them some Latin.
Oh, yeah.
They were big on Latin.
My botany professor talked about the fact that he had to write his dissertation again in Latin.
Yes. Until 2012, every new species description of a plant had to be written in Latin.
Yep.
2012.
So you'd write it in your language to be published and given out, but then you had to officially make a copy in Latin.
In Latin.
Yeah, that was my reaction when he told me that.
Nobody loves Latin like botanists do.
That's silly.
Yep.
Yes.
Yep.
Fortunately, that is no longer the case.
Well, great progress.
Thank goodness.
Thank you, Allie. We will include those links to those in the blog post. Speaking of which, every episode of the podcast has an accompanying blog post on our website. You can find that link in the episode description. The blog post will have pictures of some of this cool stuff we're talking about. We'll talk about different shapes of leaves. We'll get some pictures of Getacean, as long as they're like free use. Or if Allie has pictures of Getacean
I do.
that you want to share with us, yes, that would be fantastic. Check that out. There will also be links and stuff. Don't forget, this episode is coming out early July, which means that it is still Snake Month. There's all sorts of fun stuff happening for Snake Month. We've got the Discord channel open. We've got a live stream coming up. The Snakes and Crocs tier is open on Patreon, where you can contribute to us and also help to support reptile conservation. Also, we just recently put out another episode of Spotlight 2024 going on all through the year. Check those things out. Thanks, as always, to our requesters who requested this episode and all of our other episodes. Thanks to our patrons who asked their questions, our new patrons, our old patrons. Thanks especially to our top-tier patrons, Danielle the Bug Lover, Sarah May, Robert Mart, and Melissa Barksdale. If you, dear listener, would like to request an episode topic, you can go down to the episode description and find links to the topic request form, which is the number one way that we accept topic requests these days. So if you have ideas for what you want to hear on Allie's new podcast, Leave It to Allie, you can submit those to... We'll make a little collection of them, and then when you start your podcast, we'll shift it over.
Yeah.
I'll host the podcast if you want. It is a very common format of podcast, is that it's two hosts, one person who knows what they're talking about and one person who is clueless and is just asking questions. We could do that. I'll be your clueless co-host if you want.
I love questions. Let's go.
Listen, this is where ideas are born. We release episodes every fortnight on this podcast. Allie, thank you so much for joining us for all the plant episodes. This hence and in the future. And we're so glad that you got to finally have the Alliest of episodes.
It was the most Allie.
Let's leave.
Bye.
Bye.
Thanks for listening to the Common Descent podcast. You can follow us on Facebook, Twitter, YouTube, and check our WordPress blog for pictures and links after each episode. Huge thanks to our patrons whose support helps keep this podcast running and who get access to bonus goodies on Patreon. The song you're hearing is called On the Origin of Species by Protodome, which we found at ocremix.org. Thanks again for listening. We hope you'll join us next time.